She's more into molecular biology and genetic engineering technique, gene cloning, NGS, and she's also more into immunological techniques, immunohistochemistry. We now look forward to Dr. Deepthi ma'am to take over the session and quickly start off. Deepthi ma'am, you need to unmute yourself. Unmute ma'am.
Yeah, and I'm trying to share my screen also. So just a second.
Ma'am, one more thing. If you
Yes.
can magnify your slide, okay, that also would be better, because yesterday in viewing we had a little problem.
Oh,
So if you
okay.
could zoom, increase the percentage.
Okay. Okay.
Yeah.
Yeah. Do you see the screen now?
Yes, ma'am. Yes.
Okay. We'll start.
Sure.
Good morning, everyone. It's good to be back. I hope yesterday's sessions were great and everybody knows now what a PCR involves. Because now it's going to be a little more technical, where we're going to get into quantitative PCR and its applications. So, the storytelling I will try to do, but we'll probably need a little more focus. I'm glad that the talk is early in the morning and everybody is fresh. So, getting into Qpcr and its application, like I said, Qpcr means quantitative PCR. See, when we do a PCR and amplify a DNA sequence, we are either getting a qualitative answer, yes or no, whether the gene is amplifying or not, or the other thing to do is quantify how much is amplifying. Okay, now the thing is, what amplifies kind of has a direct correlation to how much was there in the template. Okay, so if we start with one copy of the template, we're going to do 2 in the first cycle, 4, 8, and so on and so forth. We start with 4 in the first cycle, 4 in the template, then we're going to start with 8, 16 already, right? So the quantitation of how much amplification is happening is kind of back correlated to how much template was there. So in which way we can do quantitation of your DNA numbers. So this is what Qpcr is, and we are going to learn today what are the fluorescence techniques and what kind of experimental setups we can do in order to run a quantitative PCR, and then what kind of applications Qpcr finds. So PCR, everybody knows, there's your target, melt it out, primers, binding, extension, and then you have your DNA amplified. So starting with DNA. The other way is RT-PCR, which you're going to hear more about lot today, is you start with mRNA because you don't want the intronic parts, you reverse transcribe it into double-stranded DNA and then do the PCR. So, for example, for COVID, which is an RNA virus, you have to make it a double-stranded copy, and then you will do the PCR. So, RT-PCR test that you keep hearing about for the COVID involves RT and then PCR. Now, again, we talked about endpoint PCR yesterday, where after all the cycles, you will run it on an agarose gel and see the product. Whereas for a quantitative PCR, what we do is this exponential increase that's happening in every cycle, we are monitoring in real time. So Qpcr or quantitative PCR is also called as real-time PCR. Okay, so here's what I show is an reaction efficiency. So if we assume that there is 100% amplification of DNA happening, then after first cycle you will have 2 copies. After second, 4, 8, 16, 32, and so on and so forth in an exponential way. And after 30 cycles, you will have like a 10th power 9 copies. Okay, but after the first cycle, you have a 2 doubling, exact doubling. Whereas if your polymerase of your reaction is 90% efficient, you're not going to get exactly 2 copies. And at the end of it, you will end up with only 1.9 times what your initial material is. Okay, and then with 80% efficiency, you're going to get 1.8 times every time the duplication happens. So the efficiency of the reaction is also really important. Now, how do we monitor whether the amplification is happening or not is using fluorescence chemistries. So ALX 350, FAM, TET, HEX, Cy3, Cy5, ROX, TAM, these are all fluorescent dyes which have absorptions at various wavelengths. And using these dyes which bind to DNA, we will in real time monitor how much reaction has happened. Okay, this is an amplification curve. I'll be showing more of these, and we will see how we see that the amplification happens. So cyber green, eva green, and some of these intercalating dyes work just like ethidium bromide. And they will fluoresce as in the particular wavelength that they absorb. Apart from these intercalating dyes, we have probe-based or very specific detections where the primers are either Tachman primers, beacons, Lux primers, hybridization probes with radioactivity or chemiluminescence, scorpions and amplifier probes, or FRET probes, which is fluorescence resonance transfer kind of probes. So Qpcr or quantitative PCR can be used in a whole lot of applications. Now, primers, yesterday I kind of mentioned that the primer selection or the primer design is very important. So when we do a conventional PCR or we're doing a cloning PCR, we usually make longer primers, and we the template region for amplification can be anywhere from 100 base pair to like 10 kb, 20 kb. People have done up to 50 kb also. However, when we do quantitative PCR, we're not really amplifying the whole gene. So what we do is we kind of make primers more towards the 3' end, towards the the 5' end, and we make primers for short targets. So for a quantitative PCR, mostly your target range will be 100 to 150 bases. We try, if we are multiplexing, we try to get similar annealing temperatures for the primers and approximately 60 degrees, anywhere from 55 to 70, 75 people use. But ideal, if you can have a 60 degrees annealing temperature for all the primers, that's great. Then forward and reverse primers shouldn't have a big TM difference. If one primer works at 55, one at 65, your reaction's not going to happen well, efficiently. The GC content for the primer is important, so that there shouldn't be self-hybridization or dimerization of the primers, and the free energy of the primers should be low, so that the reaction happens, is feasible, and goes in the forward direction. You can always use primer tree, DNA tree. There's so many primer design primers, or you can manually design primers. That's totally fair. And then perform a blast search so that you know that your primers are specific and they're not going to randomly or non-specifically amplify. So here's how the amplification plots look. It looks typically like a sigmoidal curve, okay, like a growth curve for any microorganism. There is a lag phase. You see, on the x-axis is cycles, every cycle of PCR, and on the y-axis is fluorescence. So initially, when one copies go to 2, 4, 6, 8, whatever, you're not really seeing a great lot of fluorescence. But there comes a point, see, between 10 to 20 cycles, where your fluorescence is enough for detection. And in this range, in here, like say 15 to 25 cycles, the whole amplification is happening exponentially, which means you'll get a nice rise in the curve. And then what happens over the enzyme, the product, everything gets kind of saturated. So then the curve kind of plateaus off. So this is how a typical amplification plot for a fluorescence detection of PCR works. See, the green here is a negative control, a no template control. So in this case, there is no amplification, no fluorescence, and whatever you're seeing post 30-35 cycles is pretty much your primer dimer or non-specific kind of amplification. So we design primers in such a way that most of amplifications happen between 10 to 25 or 30, below 30 cycles. Even for like the COVID testing, etc., they have a cutoff above which they will consider any amplification that happens as negative. So this is a very decent amplification curve for a whole lot of samples for the same gene. So same gene means it should amplify. If you've added equal template, it's going to amplify at the in a very close cycles. So here's this bar that we have is the threshold detection for cutoff fluorescence we choose, and whatever CT or cycles it is hitting here, where the threshold detection of fluorescence happens, is what we call as the CT value or the threshold cycle value for that particular amplification. Okay, the Qpcr analysis starts with DNA or RNA as template, and what we do is make primers for the DNA sequence, or for RNA we will do a reverse transcription, make it DNA, and then do the real-time Qpcr or quantitative analysis. Here you can see two curves. Okay, one is a red. These are duplicates, and the blues are duplicates. So what they have basically done here, for example, they've taken a FFP tumor tissue, got RNA from there, done reverse transcription, and with that cDNA, they have done a quantitative PCR for, say, a normal biopsy sample and a tumor sample. So if suppose you are looking for a tumor suppressor gene, the tumor suppressor gene is more in your normal tissue. That's why it's suppressing the tumor, and maybe it's upregulated in cancer. So what happens is, whatever is upregulated will show more mRNA for that particular gene, and the amplification will happen at an earlier cycle. You see, it's kind of inversely related. If you have more of the template, you'll see amplification or a CT value early. The amplification cycle will be earlier, whereas somewhere where the transcripts are lower or you have lesser expression, the CT values will come later. The cycle at which the amplification will happen will come later. So in the red, the same gene has been more transcripts, and the blue one has lower. So in this way, if you have two samples and you're comparing the same gene, you can do what is called as relative quantification. And most of the gene expression data in terms of upregulation of genes, downregulation of genes that we hear about are done by relative quantification in quantitative PCR. Now, here's where Qpcr can be used for what we call as absolute quantification. So now if you have only one sample and you want to know how many transcripts of or copy number of the particular DNA you have in that sample. So this is just like running your, Lowry's assay or Bradford assay. What you do is, with the known template, see here in this graph, 200 nanograms, 100 nanograms, 10 nanograms, 1 nanogram, 100 picogram, 10 picogram, 1 picogram, like that. They have taken known amounts of that particular gene and done a quantitative PCR. So you get cycle values or the CT values for each of these. So this is like a standard curve. And then when you take your unknown sample and do Qpcr, depending upon what cycle the application happens, you can absolutely quantify and say this is the number of the copy number of the DNA or the number of transcripts for RNA that you have in that particular sample. So with this background in mind, that you can do relative quantification starting with RNA or DNA, and you can do absolute quantification if you have a standard curve for that particular gene for in any one sample. So these are the kind of data that you will get from a Qpcr experiment. A Qpcr assay, if you have to plan, you have to first design. What is the design? What is the sample going to be, DNA or RNA? What primers? Oligonucleotide primers you have to design for homologous binding to amplify the target. Then you have to synthesize the oligos or the primers. You can outsource this. There are companies who will do it, and within a week you will get your primer synthesized. Now what you have to do is test oligos, which means you take a known sample, known template, and you run your primer and a PCR and run it on a gel to see that your band is amplified. Now, once your primer is optimized, you know that the primer works, then you use it with a probe or a cyber green with the enzyme and everything, and then get your amplification curves, and then analyze for relative quantification or absolute quantification, whichever you want to do. Okay, so experimental design, this is like really important. Anybody who's getting into research and planning a biological experiment needs to know the importance of replicates in the sample. So there are two types of replicates that we have in any experiment that we set up. First is technical replicates. So any reaction that we set up, you'll set up in duplicates or triplicates. People even do quadruplets if they want to get statistical data. So technical replicates make sure that for a one particular sample, you're getting the same CT values for that particular gene. And this reflects your experimental error. If there is, two of them show a CT of 15 and they amplify at 15 cycles, and one amplifies at 18, then the 18 one probably got a little lesser of your template. The replicates should be really, really tight, which shows your expertise in handling micro volumes. So if you set up the reactions properly, your duplicates and triplicates should be exactly matching, and that shows your handling of the system. Then the other kind of replicates are biological replicates, which means here's this experiment you've done twice in two different systems, with every sample being a replicate of the other. So this ensures that that your experiment is biologically relevant, because see, when you treat cells or you treat animals, if these particular animals were, say, left out in the cold in the night, their response to whatever you are doing may be different from the mice who would be sitting in the sun or at warm temperature. So the independent experiments that are done to replicate a particular setup are biological replicates, and n is equal to 2 or repeating the experiment twice with technical replicates gives you a very tight data if it is concordant. If you get matching CTs and similar results, upregulation and downregulation in both the experiments, then your data is publication worthy. You can be confident that what I'm saying is correct. The importance of controls, I can't stress more. Yesterday also I mentioned, you have to have a no template control to know that there is no contamination in your reaction. Without the template, there is no amplification. The primers are not dimerizing, they're not making random products. Then the no RT control, which means if you have only any DNA in your sample, then that shouldn't amplify. So if there is contaminating gDNA and your primers are such that they will amplify from the DNA also, you will probably get later amplifications, but it is not good. Which is why when we prepare RNA sample for a qPCR, we do a DNA treatment to make sure that it doesn't contain gDNA. And when we do RT or reverse transcription, we're making sure that only cDNA is there in the template for your qPCR reaction. And then there has to be a no amplification control where you don't put the polymerase. In that case also, the amplification shouldn't happen, and just your template and primers without the enzyme shouldn't give you a product. So all of these controls, whenever you set up a whole experiment for quantitation or PCR, you have to make sure that your negative control, positive control, no RT control, and no amplification control are all in place, so that your experimental errors are not there. You're sure that your system is working, your PCR has worked, and then your data is tight. Okay, PCR efficiency, again, like I said, you can confirm by replicates, doing duplicates or triplicates. The other way is doing a serial dilution. So here's, if you see this table, there is amplification of beta actin gene. Okay, the template in the first well is 25 nanograms. In the second is 12.5 nanograms, and in the third one is 6.25 nanograms. So you're decreasing the template in a concentration, halving the concentration. And then when you get the cycle values for amplification, you see that in the triplicates, in 25 nanograms, it amplifies at 15 cycles. In 12.5, it amplifies at 16 cycles, and at 6.25 nanograms, 17 cycles. See, as the template keeps going down, your threshold cycles keep increasing. The amplification is at a later cycle. So this kind of shows that, and if your R square is great, the correlation is great if your R square value is closer to 1. So here is how you can set up your Qpcr experiment to show efficiency of 90 to 100%, and you know that your data is good. Okay, this is kind of important. So far we've talked about amplification plots, which just show increase in fluorescence with the number of cycles that the PCR is going through. Now, what is this? In an endpoint PCR, we run the product on a gel. But on a gel, whatever is the maximum product will fluoresce the more. You might not see any non-specific bands if they are lighter. Now, in this case, because we have the fluorescence dye in the mix, what we do is we run a melt curve. We run the product from, see here, 70 degrees to 95 degrees. And what happens is, at a particular temperature, the double-stranded product melts or becomes single-stranded. Okay, so this shows up as the fluorescence increases here. This shows in the first case, you see one product, one peak showing one product. Whereas in this case, you're seeing a small blip here. Usually, a smaller blip in this temperature range is a primer dimer, which shows a little bit of primer complementarity or excess of primers in the reaction. It's not so much of a problem. But if you get 1, 2, 3 peaks, then you know your PCR is all non-specific, and when you run it on the gel, you'll probably see 2, 3 bands. So your target's not the only thing amplifying. You're getting amplification of other products also. So melt curve is to show that in your PCR you got a single product. So if your amplification curve is concentration dependent and your melt curve single, you know your primers and your PCR works well for a particular gene, for a particular target. Again, here if in the NTC in no template control, if you're getting a bump in the melt curve, it's kind of maybe a primer dimer. If you're doing a RT-PCR, you're doing cDNA to DNA, RNA to cDNA conversion, and then doing a PCR, and you get a peak in just the genomic DNA, then that's not a great reaction because you have DNA contamination in your sample. So you have to make sure that when you're doing an RT-PCR or a QRT-PCR, your RNA is clean and you're getting only a single product from your PCR. And with the help of these fluorescent dyes, it's easy to see the progress of the reaction. And with the melt, we can tell whether the product that's formed is specific or not. So here's a kind of report that you will get. All these samples, A in triplicate, B in duplicate, C in duplicate, all that. Oh no, it's all duplicates. A1, A2, B1, B2, C1, C2. All these are duplicates of a sample. And then you will get the threshold cycle values at which the amplification happens. For the no template control, see the cycle is 40, which means even up to 40 cycles, you're not seeing any amplification. Okay, so that's a good negative control. Now comes with this background in mind. In fact, I'd like to take 2 minutes and see if absolute quantification and relative quantification makes sense to everybody. And amplification curve and melt curve is clear. Are we good with these terms? Can a few of you?
Participants can unmute and answer. If it's okay, we will continue.
Yes, you can message or unmute and tell me if absolute and relative quantification and amplification and melt curves make sense.
Ma'am, I actually have a doubt in that the curve that you had shown, the threshold curve, this thing. Can you go to that slide?
The threshold curve, yes. This one, this one?
This one.
Yes.
So the line that you see, the horizontal line that you see over there, you told it is the threshold for the fluorescence, right?
Yes, yes.
So it means that this is the limit it is going to detect above or...
So this is the minimum threshold where we'll measure the fluorescence cycles. We take this as the threshold where the fluorescence is comparable. So most PCR machines have a threshold, default threshold set for, or we usually set the threshold where your background is minimized. So in this case, see, they have put the threshold to where the NTC is not detected. The no template control shows 40 CT. See,
Yes, yes.
if we put this threshold down, then you will see maybe 35 cycles or something as CT for no template control here. So this threshold is signal to noise. This is above, this is signal, and below this is noise.
Okay, okay, yeah.
So that is your threshold cycle. These are the numbers that we compare for quantitation. So at this threshold, all these samples, see, they show very close CTs for gap DH gene, the data for which I've shown 50, like 16 cycles, 70 cycles, or whatever. See here, all of them are amplifying at 17, 18 kind of cycles. There shouldn't be, see, a gap DH, or what we call as a housekeeping gene, it should not show differences in amplification, because it's constitutive and it's so abundant.
Yes.
And it's abundantly expressed. So we always use a housekeeping gene where the CTs will not vary. This is to keep in, to show that your input of the cDNA template is equal in all the samples. See, if your housekeeping gene starts varying, you would assume that in F1 and F2 samples here, there is slightly lesser cDNA, because you are seeing a slightly later CT value for this sample. Even the duplicates are good, 18.40 and 18.35. However, it is a little later than all the others, which are showing like 17 CT. So this is to normalize, or to make sure, if I take a normal sample and a tumor sample, I know that this particular gene is upregulated in tumor. However, I have put a lot more template of the normal. Then what will happen? In the normal, it will look like it is upregulated, isn't it?
Yes, yes.
Because the cycles depend on how much template you have put. Right? If I put only half the template of your cancer cDNA and normal cDNA, I put lots more input, then it's going to show upregulation. So to make sure that you're loading equal template of each, we run a gap DH or beta actin or some gene which will show equal CTs, because it's a constitutive gene and abundant. So if both the samples gap DH is same, and then you see that in tumor it is upregulated or downregulated compared to your normal tissue, that's a good quantification. We'll talk about that a little bit more further.
Okay. There's one more request here.
Yes.
They want to know about the controls that you had told.
Yes.
Can you repeat that control?
The controls, yeah, the controls are important. So see, a PCR reaction should work when all the ingredients are there in it. The template has to be there, primers, enzyme, and buffers, and everything has to be correct. Now, why we put a no template control is to test. No template means you're not adding your target gene or sample itself. So this shouldn't show amplification, which means your enzymes, your primers, and your other stuff is all clean, and you're not getting any primer dimers or any non-specific kind of amplification from the reaction mix. Okay, then, and a no amplification control goes in without the enzyme. So here you're putting template and primers and everything, you're not putting the polymerase. So even then, without the enzyme, the amplification shouldn't happen. And if your primers are non-specifically annealing with the template and all, you might get slight fluorescence, but it won't be like a sigmoidal curve. So you're sure that there is no background or random binding of primers, template, or dimerization and things like that. The important bit when doing gene expression studies is you want to study RNA transcripts. So either we design primers which will amplify only the exon, intron, things, so that there will be no amplification from genomic DNA, or else we test a no reverse transcription control, where you are putting your RNA as such as a template. If RNA shouldn't really amplify, in a qPCR reaction, unless it is converted to cDNA with reverse transcription. But if you see amplification in that kind of a system, then you know that there is some genomic DNA contamination in that RNA, which is amplified. So then your comparisons are not that great. You have to make sure that it's only cDNA which is the template, not genomic DNA. So these are the kind of controls we put whenever we're studying gene expression for upregulation and downregulation in a quantitative system. So most of the time we'll be doing relative quantification. We have, treated, untreated sample, normal tumor sample, or mutant, or wild type sample. In both of the samples, we'll put an equal template and then see whether the particular gene that we are doing the qPCR for is upregulated or downregulated. Cool?
Sure, ma'am. Yes.
Okay, so now coming to, if you understand the amplification curves and melt curves, we can go on to applications of PCR. So the simplest thing to do is gene expression studies. I already kind of told you an example of a normal tissue and a tumor tissue, or a, untreated cells versus different concentration of your drug treatment, or toxicity kind of experiments you can do, gene expression in wild type versus knockout. All those things can be done one gene at a time. You put just 2 reactions to compare. You're doing one gene. You can do, in an 8-well strip, you can do in a 12-well strip, you can do in a 96-well plate, a 384-well plate, depending upon how many reactions you want to set up in one go. The various cyclers will do as many reactions. The various thermocyclers that you can use. Now, where does this gene expression studies come into use? When you are, you want to study one gene, 10 genes, 50 genes for their expression. There are, in fact, later in my slides, I have what we call as Qpcr arrays. So these are ready-made. They are from super arrays. They were taken by QIAGEN now. They are 96 well plates with primers already put, and the reaction mix, everything put in each well. You just have to put your template cDNA and run the qPCR, and then it will tell you whether the particular genes amplifying in that sample or not. So inflammatory genes or apoptotic genes or cancer related genes, there are different gene expression arrays that you can run. Okay, when you do a microarray experiment, is everybody familiar with microarrays? DNA or RNA microarrays?
No, ma'am.
Uh-oh.
No, ma'am.
So
No, ma'am.
microarrays are kind of fishing expeditions. You have probes for say 1 million targets on these slides, and then you hybridize your sample to see which and all transcripts are binding to the probes. Okay, and microarray data comes out as spot intensities. So you will, when you do your genomics curriculum, in there you will study about microarrays. So microarrays detect at a time the expression of say even 1 million targets. We have about what 35 to 40,000 target transcripts in our transcriptome, but they'll make multiple probes for for every gene, and at a time million of templates can be tested. But see, microarray data is hybridization based. So you will get out a whole lot of data for these million transcripts, whether they are upregulating, downregulating. But in order to confirm or actually validate whether this expression is correct or not, you will use qPCR. So out of say 1 million genes, you see that there are these particular 50 genes which are showing really upregulation in a particular sample. Then what you will do is you will design primers, you will do the whole cDNA preparation and do qPCR to validate whether what the data that you got from microarray actually verifies in a qPCR, quantitative PCR. So the first two are kind of related. Gene expression is independent qPCR, whereas validation of microarray data, you will choose which genes are showing good regulation, upregulation or downregulation in the microarray hybridization experiment, and then confirmed by qPCR. The third application that we're going to study is Snp genotyping. I hope everybody is familiar with single nucleotide polymorphisms, which means a one base which is flip-flopped in the two alleles. So that kind of studies we can do using qPCR. DNA methylation analysis. So we do the bisulfite. We talked about pre-treatment yesterday. So you can do, you can study the DNA methylation status by doing a bisulfite treatment and then doing qPCR for comparisons, whether hypermethylation is there or hypermethylation is there. You can do those kind of analysis using qPCR. Then for diagnosis, everybody knows the qRT-PCR for COVID. So that I don't even have to go into. And I think the next talks today are going to talk more about RT-PCR and COVID testing and things like that. Okay, apart from your mRNA transcripts, we also have a whole lot of other RNAs that are there now: long non-coding RNA, siRNA, snRNA, hnRNA, and miRNA, the microRNA, which have also been shown to have a lot of regulatory role in a lot of diseases and different statuses. So nowadays people are studying upregulation of these microRNAs by quantitative PCR. The thing about microRNAs is they're micro. So they're about what, 20 to 25, 18 to 25 bases. So you have to do it in a, you have to do the qPCR in a very different way, because the primers that we make for qPCR for mRNA are in the range of 18 to 20 bases, isn't it? So the miRNA primers or probes have to be a little different, because miRNA itself is about what, maximum of 25 bases. So miRNA profiling is another field where qPCR can be amplified. So basically I was going to go through all of these in a little detail. Please bear with me. We'll go topic by topic, and if you have any doubts, I'll take questions in between. You can unmute and ask, or at the end of all the techniques. Okay, so yeah, analyzing the gene expression data for relative quantification. It's called the delta-delta-CT method. It sounds complicated, but it's simple. So the delta-CT, or the change in CT values, is first calculated for the target gene by subtracting the housekeeping gene. Like I said, housekeeping gene CT values should not vary because it's constitutive and abundant. So this normalization is simply done to normalize that all the templates for all the samples are equal. So the first is your normalization, data normalization, by subtracting your cycle, threshold cycle for the GAPDH or the beta-actin gene from your P53 or whatever target gene that you are studying. The second delta that we do is your comparison. So experiment sample minus reference sample, your cancer sample minus normal tissue, or knockout versus wild type. That is your delta-delta-CT. So the delta, these, you're simply subtracting the CT values. The delta-delta-CT method sounds very complicated, but it's really simple. And once you have this delta-delta-CT, this can be exponentialized to by using this formula in order to get you fold expression, fold up-regulation or down-regulation. I'll show you with an example. Okay. So here's data from for two genes for a control and a treated sample. Say normal and tumor, if you will, control and treated. So first gene, a gene of interest, is showing a CT of 20. At 20 cycles, the threshold is crossed. For the treated sample at 23 cycles. Okay. Then second gene, 22 cycles, the control shows amplification, whereas the amplification in treated sample is at 18 cycles. And then you see 18S RNA, which is again for normalization, your constitutive or housekeeping kind of gene. You will see that both of them are showing 18.14 and 18.18 as their cycles. So this is a good data where your housekeeping is not showing any differences in CTs, whereas your genes of interest are showing different CTs. So the first delta CT that we do is simply subtracting the value for housekeeping from your actual data. So here this is 20 minus 18.14. This is 23 minus 18.18. Okay, that is the cycle difference for the gene of interest 1. Here 22 minus 18.14 is 3.86 and 18 minus 18.18 is minus 18, which means your gene of interest 2 is amplifying even before your housekeeping gene. So delta CT just is done to make sure that equal loading of sample was done by subtracting the housekeeping gene. And now comes your delta delta CT. In the delta delta CT is where we are comparing the control versus treated. So you subtract 4.82 minus 1.86 gives you 2.96 here. So you know that the difference between your treated and control is 2.96 cycles. Okay, and in the case of the second gene, it comes to -4. Now, what does this mean? If you actually look at the delta delta CT, this means treated samples getting a later CT, which means your gene is down regulated. It's amplifying later. Whereas if you get a minus value for delta delta CT, your treated sample is showing earlier amplification of the gene compared to your control. So this, even by this plus and minus, you can tell upregulation, downregulation. However, when we plot it for graph or we plot it for a publication, what we do is we convert these delta delta CT values into exponential form, which tells you fold change. So this 2.96, when you do a 2 exponential minus delta delta CT, you get a value of 0.04. And if you do for minus 4.4, you get 16.45. So any value more than 1, for 0 the value comes as 1. So if you take 1 as your housekeeping, compared to that, your gene of interest 1 in treated sample is 0.4 folds, which means is even less than half. Is like what? Yeah, it's less than half. Whereas the gene of interest 2, because it's showing an earlier CT value in the treated sample, is 16 folds upregulated in the treated sample. So values more than 1 show upregulation, and your graphs will go up. Whereas a downregulation, the graph will go down, or it is from the 1 threshold, it is lower. So that's how we plot. Either you can color code upregulation, downregulation, upregulation, or you can make a bar graph which shows upregulation and downregulation, and that's how you represent a quantitative data in a report or in a publication. Okay, so basically what happens is what you get from microarray data and from Qpcr data is confirming the upregulations and downregulations. Okay, both of them have different kind of how you look at the data. So in microarray, you're looking at spot intensities and you're looking at upregulation and downregulation. Whereas in Qpcr, you're looking at CT values. So it can't be exactly, if your microarray is showing 10 folds, it's not necessary that your Qpcr will also show 10 folds increase. But if your trend is similar, then you know that the data correlation is there. Here, see, if you see this data, the top one is showing microarray data. Of a blue and green comparison. So here upregulation, 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 all of these genes are upregulated in the green. Okay, so but what happens is when you do this by Qpcr, you may not see the same degree of fold change. And see, in some of the samples like this one here you're in the microarray, you're seeing upregulation, but in the Qpcr there is not so much fold change. But still it is the green is little more than the blue. So what happens is, even in the microarray data, the more difference or the fold change degree of change you have in the microarray data, the more likely that it is going to validate with Qpcr, which is why for Qpcr validations of microarray data, people choose the highest upregulation or highest downregulation kind of genes. If you choose genes which are only little bit upregulated, little bit downregulated, they may not validate that well by Qpcr. So this is one of the pitfalls of validating microarray data. And also the direction of change. For upregulations, the validation is much better than for downregulations. So with these things in mind, if you pair your microarray data and qPCR data, even though they require, different normalization procedures and the things, but it has been demonstrated that they should yield comparable results. And whenever you do a microarray kind of experiment, you have to do qPCR validations for the genes to confirm that what you're getting from the microarray data is not by fluke, and you're actually seeing upregulation or downregulation of those particular genes. So like I said, qPCR as such you can do independently, or you can choose genes which are shown to be upregulated or downregulated in a microarray data and confirm them by running a qPCR experiment. Quantitative PCR will tell you upregulations and downregulations. Okay.
Thank you, ma'am. Yeah.
Yes.
Related to the earlier one,
Yes.
there is one question that I would like to ask you.
Sure.
Raised by a student.
Yes.
Okay. So, how to determine genotypes in viral genotyping? Can we determine by using these curves after amplification, or do we have other methods?
Absolutely. The next part is Snp genotyping for genotypes. So since so far we've been talking about transcription, we've been talking about gene expression, mRNA quantification. Now coming to your viral or single nucleotide polymorphism genotyping, in if you have a wild type versus a mutant, or you have a virus which has various genotypes, we can use quantitative PCR. Okay, how it is used is either by using a high resolution melt curve, by Tachman probe methods, or by a chemistry called Casper chemistry. We can go into this also, but it's these are all a little technical, so focus. By high resolution melt, you remember the melt curve? What we are doing is we are melting the amplified product, and the temperature at which it is becoming single stranded is kind of giving you a fluorescent peak. So now think of it as you have a homozygous kind of allele, and you have a heterozygous where there is a SNP or a polymorphism. The polymorphic one will be lesser complemented, isn't it? So what happens is you get when you do a high resolution melt curve, you're going to get a earlier temperature denaturation or melt peak for heterozygous versus homozygous. Does that make sense? A high resolution melt curve to tell whether the polymorphism is there or not?
Yes, ma'am. Yes. We have got response. They have understood.
Yes. So basically, in a high resolution melt, what do we do is we can analyze genetic variations in a PCR amplicon, or what you are calling as mutants or polymorphisms. Okay. So here what we do is we don't run a classical melting curve. There is in the qPCR machine what we call as a high resolution melt, which means not just melting it from like 70 degrees to 90 degrees, it will do 70, 71, 72, it will do a higher resolution kind of melting and measure the fluorescence accordingly. And then this thermal denaturation of the double-stranded DNA gives you a more information for whether both the strands that are amplified in the product are homozygous or there are polymorphisms there. Okay, so it pretty much uses the same principle as the dissociation melt curve, and a single base causes a different melt curve because the DNA is not as homologous in the heterozygous thing, and it will melt at an earlier temperature or a lower temperature than your homozygous. So a lot of genotyping for hybrid plants, for knockout animals and knockout microbes can be done by using high resolution melt. So from the shape of the melt curve and the temperature at which the DNA will dissociate or melt, you can tell whether it is homozygous or heterozygous for a particular position. So that's how you can do genotyping. So basically, what are the advantages of using high resolution melters? It is, you can do it in a regular quantitative PCR where we are using cybergreen or evergreen and the primers. You don't really need a special instrument. You don't need anything special. You run your PCR, Qpcr, and you have to just run a simple high resolution melt curve at the end and identify your SNPs. Versus this is the more abundant or abundantly used or more popular kind of method. It's called Tachman probe based method. So here your probe or the primer, I mean we call it a probe because it's tagged with fluorescence, and this uses for SNP genotyping. How it is used is the probe for wild type, and there is one probe for the mutant or the polymorphic variant. Okay, and then the probes are labeled differently, either with Psi3, Psi5 or FAM, TET, hex, whatever. The fluorescence for both the probes is different. Okay, I'll show you a figure. Hopefully this will make it more clear. So here in the first case, there is a the base at this position is G, and your probe has a C, which means the probe will bind, there the PCR will happen. This will release your reporter, which is fluorescent, and you will read fluorescence in the amplification. Okay, here this means the PCR is working. Whereas for this case, because your base is G and not A, the mutant probe will not bind, and the other reporter will not work. So depending upon whether the Psi3 or the Psi5 lit, you can tell which of the bases is present, whether it is G or A. Okay, so Tachman assay is designed in such a way that you have probes for the wild type as well as the polymorphic base. Whichever binds will release that reporter, because both the reporters are different fluorescence, and you can tell whether you have the wild type or the polymorphic allele. So Tachman probes are a little more expensive, but if you're doing say 100 samples, then it makes sense to make both the probes. Because these probes come with fluorescent tags, the primers themselves, or the probes we call them, come with fluorescent tags with a quencher and a reporter. These are like what 4 or 5 times more expensive than the regular primers that we synthesize. So Tachman assays are very, very specific and very, very abundantly used for detection of the variants or mutants or SNPs, whatever you want to call them. And I mean, you can use them in a high throughput way. So they are very highly specific and fluoresces only when your specific amplification is happening. So you have a high signal to noise ratio. I mean, whether this is working or that, it is very, very clear. And you can put multiple probes and do multiplex reactions for various SNPs in the same template. So Tachman probes are a good way of doing genotyping. One more method is called as a Casper chemistry method. Again, instead of reading this, I think I'll go to the diagram first. Here, what happens is you have one reverse primer, and the probe itself is such that either it is for this or this, and based upon which one amplifies, you can tell whether it is a wild type base or a mutant or a polymorphic base. So in Casper chemistry, you have 3 primers. The reverse primer is common, whereas the forward primer is right where you want to detect the mutation. And then depending upon which of the forward primer binds, you can tell which polymorphism is there. Okay, so these are the 3 ways to do polymorphism or variant studies. The question at the beginning of this part was for viral mutations or SNP detections, how can Qpcr be used? So this is the way, either by using high resolution melt, or by using Tachman probes, or by using Casper primers, which are fluorescently tagged. You can figure out which polymorphism is there in which allele is present. Does that answer your question?
Yes, ma'am.
Okay, so yeah, pretty much the advantages of Casper are that it is robust, and because you're using primers which are closely tagged, not like Tachman probes with a quencher and everything, they are much cheaper, and you don't need probes. So every method to do SNP genotyping, whether it is high resolution melt or Tachman probes or Casper chemistry, has its own advantages and disadvantages, and depending upon how many samples you're analyzing, how many qPCRs you'll run, you choose your system. I already spoke about PCR-based microarrays for gene expression validations. If you want to do a particular set of genes, you can have primers for all of them, and you can even customize. In a 96 well plate, you can have primers for, like, say, all inflammatory genes or all apoptotic genes or all necrosis-related genes or whatever you want to study. And you can do qPCR at the same time and then analyze the data. So these are supplied by Super Array Biosciences, which is now supplied by QIAGEN. So if anybody wants to do qPCR validations of known systems, you can use PCR-based arrays. So in clinical laboratories for infectious detections, multiple probes for multiple viruses at the same time or multiple pathogens, you can use. And these are all the regular intercalating dyes like cybergreen and evergreen based super qPCR. The last bit, the last 5 minutes of my talk is, like I said, miRNA profiling. MicroRNA is actually new, because still new, because till 2 years back we had only 700 microRNAs known. Now we have more than 1,000 microRNAs that have been known and profiled and associated with targets or what they regulate. Some of them have been associated with various diseases, the upregulation of a few microRNAs with correlated with diabetes, with particular kind of cancers and things like that. There's so much research going in the field of microRNA. So, like I said, the thing about microRNA is it's really small, 18 to 25 bases. So what is done for qPCR is, first you add a poly A tail to a microRNA, so it becomes like an mRNA, okay, and it becomes longer. Then your reverse primer is in that poly A tail. You do a cDNA synthesis for this, so it becomes double-stranded, and your universal primer is for your poly A part. And only one microRNA specific primer is what you need for this amplification. So the only challenge for microRNA is that it is small. So the strategy is easy. You just put a tail in such a way that you can have a universal reverse primer. And you buy or synthesize only one forward primer which is specific for your miRNA and do the qPCR. It's a simple strategy. So the advantages are, you can do microRNA arrays also, but it is faster. This one is faster, and you need very little amount of your RNA sample. However, now I've said over 900, but there's more than 1,000 miRNAs now known. So if you have to analyze all of them by qPCR, it will take a lot of your time. And the miRNA primers also, again, because they're very specific, they're more expensive than your regular primers. So miRNA research is slightly more expensive than doing transcription or gene expression kind of studies. And so here I have kind of chalked out the strategy in miRNA kit, but then CloneTech, Introgen, and there are other miRNA analyzing qPCR kits also. Basically, they have different forward primers for different miRNAs and slightly different system to kind of detect it. But I mean, technically, you are doing a tailing, cDNA synthesis, and then a regular kind of forward reverse primer kind of reaction to quantitate your miRNA in the samples. So with that, I hope the talk about qPCR, or how we quantitate in an absolute manner or a relative manner by checking out the fluorescence curves, amplification curve and a melt curve tells you specificity of your qPCR. I hope everything is clear about what qPCR means, quantitative. And then the kind of applications are for doing gene expression or miRNA expression, SNP genotyping. These are diagnostics for say COVID and things like that. All of these can be done using qPCR, because you can monitor in real time whether amplification is happening, how soon the amplification is happening correlates with how much more input template you had. Viral load can be estimated like that. If you have more virus present in your sample, the CT values will be really early. So which is why for like even a COVID test, you have a cutoff. If your if amplification is happening say beyond 30 cycles, then it is a negative test. So if you see viral gene amplifying in say at say 20 cycles, you know there is plenty viral template in the sample and the person is infected, is positive. So in diagnostics, in gene expressions, in mRNA expression, and for variant studies, polymorphism studies, you can use qPCR. If there are questions and doubts, I'll be happy to answer them.
Yes, ma'am. There's one question.
Yes.
Are heterochromatin DNA detected in PCR? Heterochromatin.
Sure. I mean, if you know the sequence and you design your primers and probes in such a way that they will amplify that region, of course you can.
But in what kind of studies do you think it will be significant, ma'am? Heterochromatin?
See, when we in 2003 when we came up with the first human genome, we thought, wow, by there was this whole talk by 2020 we'll have everybody's genome sequenced and we'll have personalized medicine depending upon what the genome of the person shows, what kind of susceptibilities to diseases, what kind of drugs they'll respond to. We thought we will decipher everything about a human genome. We are anywhere close to deciphering the whole genome. Even the exome part, which means only the coding part, which is like 2 to 3% of the genome, they have kind of figured out and correlated with markers for various kind of epithelial cancer or colon cancer or whatever, or diabetes or cardiovascular diseases or whatever. So there are panels, and I mean there is progress, yes, but the heterochromatin part, because there are so many cis and transacting elements, regulators, the 3D structure of the DNA, we don't know. I mean, we figured out one stretch of sequences for the whole genome, but we don't know how they fold, how they interact in a trans manner, what regulates what. If the genes on one chromosome affect the genes on other chromosomes, nobody knows. I mean, there is no a total understanding of how the heterochromatin works. So yes, there are studies going on where they study the effect of regulatory elements or repeats and stuff like that in the heterochromatin, which can be studied by qPCR. Yes, but we are nowhere close to knowing all the mysteries of the human genome at all.
Okay, ma'am, one last question I would like to take, because we
Sure.
will slowly moving into the next session now.
Sure.
Can quantitative PCR be used instead of RT-PCR for more efficient diagnosis of viral diseases?
So here's the thing: if your virus is HIV or COVID for that matter, and it's an RNA virus, how will you do PCR without making it double-stranded? You have to do reverse transcription, right? Because a PCR amplification happens on a double strand. The forward primer binds to one strand, the reverse primer binds to the other strand, isn't it? There are now polymerases which will amplify direct from RNA, but that also in one step. There is a one step where RT reverse transcription happens and then PCR happens. So you don't have to do reverse transcription separately and the PCR separately. You can do it in one tube, but you can't directly use RNA template for quantitation. So far, that's not a very popular kind of option.
Okay. Thank you, ma'am. So I would like to thank Dr. Deepti ma'am for giving a valuable time for both the days. Okay, you have addressed
It's my
all the
pleasure.
types of PCR, and today you had addressed about the quantitative PCR and its
Yes.
advantages in gene expression or for SNPs or in diagnosis or
Yes.
microRNA analysis and all those
Absolutely.
things. You
Yes.
have really addressed and thrown a lot of light into all those things. Wonderfully, you have depicted it with experimental studies. Thank you so much, Deepti ma'am, for taking up your precious time and being part of our workshop and being a valuable resource person for this. Thank
Thank
you
you.
so
I'm happy
much.
to be here. Glad to connect with students. And like I said, if I can, inspire even a few more to get more interested in biological, biotechnology, research, lab work, my job's done.
So sorry for the questions which has not been raised. You can kindly mail it to Deepthi ma'am. You have her mail ID which was being provided. Ma'am, if you
Yes.
can give your mail ID again.
Yes. So it's still presenting, right? I mean, you still see my screen, isn't it?
No, no, ma'am.
Oh, it says it's still presenting. Hold on. Here we
Yes.
are. Yeah, you see it now?
Ma'am, if you could enlarge it. Yes.
Yeah.
So you can take down her contact number and mail ID and you can
Phone
post
number,
your
mail
query.
ID. You can WhatsApp me. You can email me. I will get back with your answers.
Yes. Thank you so much, ma'am. Thank you. Thank you
Have
for
a nice
joining.
day. Enjoy your workshop and learn.
Thank you.
Okay. Bye bye.
Sushil sir?
Yeah, I am waiting for sir.
Okay. Sir is yet to join, right?
Yeah. I'm waiting.
Okay. Okay.
Give just 2 minute break to them.
So you have 5 minutes break. You all can join back sharply at 11:15. Sir,
Good morning,
sir
sir.
is joined.
Yeah. Good morning, sir.
Good morning. Good morning.
How are you, sir?
I'm very good. Very good. All
It's
right.
nice to see you, sir.
Thank you. Thank you. Same here. Same here.
Sir,
Thank
just 2 minute students will join, sir. उनको break दिया थोड़ा.
you. Okay. Okay. All right.
Thank you very much, sir.
Thank you.
Emma, ma'am, can you message others to join back?
Yes, sir,
Emma,
I'll do. Yes,
yeah,
yes,
please,
I'll do.
please message.
I think the title that we have given, no, in thinking back, I think we should have given some little bit more broader title. Anyway, my talk is going to be a bit broad, but that also sometimes students, they obviously just PCR doesn't work for them.
Sure, sir. Thank you very much, sir. No problem, sir.
But anyway, I think.
आप जो भी बोलोगे इस अमृत सर.
Sorry. No, that's kind of you. Well, I'll just do a broader talk, a little bit broader than just PCR, to make a little bit of story around it so that
Sure,
it's
sir.
more interesting.
It's always great listening to you, sir.
No, thank you. Thank you. These are mostly master students or?
Sir, it's a mixed gem, sir. Like students from graduate also there, masters also there, and faculties also there, sir.
Okay. Good, good.
So another 30 more has to come, sir. So after that we will start. Close to 160, 170 is the population.
Okay.
Sure, sir.
No problem. Take your time.
Thanks, sir. Just 1 more minute. आपने क्या स्क्रीन शेयर करनी है सर? Will you share the screen, sir?
Yeah, I can share the screen. That's
okay,
okay.
Sir. Here down is okay. Sharing is there, sir.
Yeah.
In present now you can share it, sir. Down.
Can you
is,
see?
No,
Can you
sir.
see my screen?
Not yet, sir.
1 second. Yeah, now you can see it.
Yeah, we are seeing, sir. Yes, sir.
All
The screen
right.
is visible, sir.
Yes.
Yes, sir.
But should I put like a small window or should I put the whole screen?
It's better to put the whole screen.
Okay.
If you want, you can just F5 and you can continue. But don't close the screen, so it will be opening. If window you do it, then you have to open the screen and leave it. If you do just screen, then directly you can showcase.
Okay.
Yes, sir. Slideshow कर दीजिए.
1 second. There's some issue coming.
No problem.
Is it there now?
Yeah, it's yes, sir. Screen is there.
1 second. Yeah. Now this should be full screen.
Yes, sir. Yes, sir. Full screen, sir.
Okay.
Yes,
And I have about 45 minutes to 1 hour, is it?
yes, sir. So, Hima ma'am, shall we start?
Sir, participants have joined.
Yeah, it's already 164 now.
They will join, sir.
So they will
We
keep
can
on
start.
joining.
Yes, we can start.
Okay. Good morning, one and all present here. So I'll start with some quotes. In science, the credit goes to the man who convinced the world, not the man to whom the idea first occurs. Impossible only means that you haven't found the solution yet. And the person who is sitting in front of us, today I'm going to introduce a person who is dreamy but less spoken. He is Superman of proteomics in India. I know him a little way back. So a man behind One Health, concept for global health initiative. Let me introduce the man. For me, his mentor. His name says Utpal, and in Hindi, it says Kamal. Extremely rare. Same way, his talent is also rare, and can be said rare to find. So Utpal sir, welcome you. I just introduce you with few lines. Dr. Utpal Tatu obtained his PhD from Indian Institute of Science and did his postdoctoral PDF postdoctoral research at Yale School of Medicine, New Haven, Connecticut. He is currently a professor in Department of Biochemistry at Indian Institute of Science, a Makkah of India, a Makkah of science, a Makkah of learning. His research aims to develop cheaper and more effective medicines for neglected diseases. So to name few, malaria, trypanosomiasis, amoebosis, Professor Tatu has made extensive use of bioanalytical tools, including mass spectrometry in his research for both proteomics, metabolomics applications. So that's why I said he is Superman of proteomics. He serves as an expert panel member on United States Pharmacopoeia Therapeutic Protein Panel. In addition to carry out educational workshops for various institutions, he has offered thematic workshops together with the United States Pharmacopoeia on bioanalytical methods for various biopharma companies. Professor Tatu's lab has established a successful academia-industry interaction. In addition to analyzing over 2,000 challenging samples for various biopharmaceuticals and pharmaceutical industries, his lab has collaborated with various national and international pharmaceutical industries for drug development projects, biosimilars, characterizations. Professor Tatu holds international patents and is a recipient of several national and international awards, including Birla Science Prize, Bioscience, Career Development from DBT, Ranbaxy's Research Award, to mention a few. He is an elected fellow of Indian Academy of Sciences and serves on the research advisory board of several biotech companies in India as well abroad. He is closely involved in advising biopharma industries, biopharma companies, and owns one equine. So Professor Tatu is an editorial board member of several big journals, from one among of them Parasitology, published by the Cambridge Press. He serves the President of Proteomics Society of India and was formerly Vice President of Indian Society of Mass Spectrometry. So thank you very much for accepting our invitation, sir. I will now, Mike, I will give to you. Thank you, sir. Yes, to you.
Thank you, Dr. Sushil, for your kind, generous introduction. I'm projecting the screen, but am I also visible in a window or not? Only
Yes,
the
sir,
screen.
you are visible, sir. You are
Also.
visible, sir.
Okay. All right. Okay. Thank you, Dr. Sushil, again for making me part of this wonderful workshop and such a nice initiative you've taken for the benefit of students, and at an appropriate time when these methodologies, PCR, has become so relevant in human life and human survival, I would say. So it's a very well timed workshop that you've organized, and very well thought out topic. And I saw that you invited so many people from different backgrounds, including some diagnostic companies which shared with you some of the COVID-related applications of RT-PCR. It's always, I'm no stranger to Ammanni College. I've been talking to various faculty, the honorable the principal. I've had many opportunities to interact with Ammanni College, and it's always been a pleasure for me because we are just so close to Indian Institute of Science that it's a delight to have students visiting from Ammanni College who are inquisitive, who are motivated to learn more about research. So that's why I put this first image of the Indian Institute of Science campus. And as you can see, Ammanni College can probably be traced somewhere in the vicinity here. Probably it's just cut off in this picture, which is an aerial view of IISc campus. And I would urge all the students who are attending today's talk that they should come and visit the Indian Institute of Science, come for a walk, and also visit our departments to understand what's going on. And it'll be a pleasure. We always have opportunities for desirous students who want to learn more things and apply science in various different ways. So it's an open invitation to any student who wants to participate in our endeavors. And I'm going to talk to you a little bit about, give you snapshots of what we do in my lab at Indian Institute of Science, which I hope will attract you to come and think about doing some more work with us. So again, I'm going to the title of my presentation today is going to be, of course, about PCR, but more about PCR in academia, industry, and community service in the broad area of neglected diseases. As Dr. Sushil mentioned, I've spent most of my career for the last 25 years studying infectious diseases, and particularly neglected infectious diseases, which are common in countries like ours and developing countries and also underdeveloped nations. But now, of course, it's become relevant for the whole world in the time of COVID pandemic, because we have become aware that infections are not necessarily limited only to underdeveloped nations or developing nations, but they can be very common also in developed part of the world, Europe, Americas, and whatnot. And so it's an important topic that we must understand. I have divided my talk into some sort of titles, but I think having looked at the program, I think this audience probably does not need introduction to PCR, but because many other speakers would have covered introduction to PCR already. So basics of what is PCR would already be there with students, and I wouldn't want to repeat it. But what I'll do is I have tried to create a story around use of PCR in our own research, which is centering on this concept of One Health and neglected infectious diseases, with some focus on some infections that I'll briefly mention, but then again talk more extensively on COVID, about how a scientist can play a role in the times of COVID. And then finally, I'll wrap up with some other interesting applications of polymerase chain reaction. And of course, needless to say, there will be question and answer session at the end where you are, I'll be happy to answer some questions, because it's impossible to cover this vast area of PCR, but I've tried to make it like a story because many of you have already heard about this thing. And as a result, I will apologize and skip a couple of initial few slides, because they must have been common among many other speakers, because most of the speakers are actually focusing on these aspects in one way or the other. So let me switch directly. I would have shown you some basic introduction to PCR and then some applications, but I will jump directly into the main topic. Right, so what is One Health? And that's really where I will start. And what are neglected infectious diseases, and how we can apply this concepts of polymerase chain reaction here. What I'm showing you here really is what my research laboratory at Indian Institute of Science endeavors to do. We, as many other professors at Indian Institute of Science, I too focus on a specific area, and I focus on infectious diseases with a focus on basic understanding through multidisciplinary tools, not limiting to a particular technology, but as the question demands, we apply technologies ranging from basic biology, cell biology, to omics sciences, genomics, proteomics, metabolomics, and then systems biology. And then, of course, as most of you know, we publish papers, and that's really one of the most important mandates, that we make research contributions through visible publications. But over the years, of course, I have made an effort that apart from fundamental understanding of infectious diseases and their molecular pathways, can we do something for the community? And over the years, then I've sort of started working with clinical doctors in both in human health and animal health, and worked with clinical samples coming from animals and humans and applying many of these omics technologies and basic aspects to be able to ask that what we do in the laboratory, is it also relevant in the field? And that's something that I've derived a lot of satisfaction in. And over the years now, we have also moved one step forward and tried to ask that, can we now, the science that we have done, can we actually develop that into some useful tools? Useful tools which can be applied in the community for the betterment of human health, animal health. And that's really where I am at the moment, that our science has really reached the community. And it has been very satisfying journey where we have not only developed good, useful diagnostic tools, but created very useful drug targets and candidate drugs, which are being considered for drug development. And in this journey, the reason I'm mentioning this is for students to know that today, being a scientist doesn't necessarily mean that you are teaching and you are merely teaching and you are publishing papers. This was the classical view of a scientist at IISc, but today scientists are moving on and not doing work with hands in hand with clinical personnel, doctors, and various other engineers to devise new technologies. And then, of course, many products are actually coming out, wherein scientists are playing also the role of entrepreneurs. They are able to actually not only do their own research, but also able to start platforms, start companies wherein products can be generated through the research that they've done. And I'm one such example from Indian Institute of Science, where the institute has empowered me with and had the confidence to allow me to also do more than just research limited to the laboratory, but much more in terms of its applications. And this is what I'm going to talk to you today about, how my research on infectious diseases and in fact neglected infectious diseases, because many of these names, many of you may not even have heard. For example, trichomonosis, trypanosomosis, babesiosis, whereas other names such as rabies, malaria are more familiar to you. I'm going to explain to you what are these terms, what is meant by neglected infectious diseases and this concept of One Health. This slide is particularly relevant before I jump into applications of PCR, because what this slide shows you is that in infectious diseases, it turns out that more than 70% of the infections that we face today, that humans suffer from, actually come from other animals, birds, and reptiles. In fact, we are living in an ecosystem which is shared, which is co-shared by all the other living beings. And so problems that are encountered by these animals and these birds are also become problems for us. And that's the new awareness with which I have conducted my research. Over the last 10 to 15 years, I'm looking at not only human health, but also birds, animals, and many other creatures in the world, because today, this is very well borne out by COVID-19, which all of us know appears to have jumped from bats into humans and through some intermediate animals. So, and this is exactly what I would like to discuss today, and how PCR plays an important role in this aspects. Just to show you again this point that being highlighted, that most of the outbreaks that we have faced in the last 10 to 15 years, be it Ebola, Zika, swine flu, avian flu, and then most recently COVID-19, Wuhan coronavirus outbreak, have actually all come not from species such as bats, other animals such as pigs, camels, or poultry into humans. And so this is really what I think a new awareness that all of you as scientists and budding scientists must keep, that we must try to take care of animals, and enough emphasis must be put on monitoring animal health if we want to remain healthy and away from infectious diseases. PCR, of course, has played an important role in this. And I'm going to start by giving you some real life examples of the research and the contributions that we are making from my laboratory at Indian Institute of Science. As I mentioned in my first slide, my research career at IISc, every few years, I have tried to incorporate one new topic. I started my career working on malaria in 1997 because at that time, malaria was a very important problem. It was causing significant morbidity and mortality in the country. Today, of course, we have done significant research contributions in the area of malaria, including, of course, identification of novel drug targets and understanding various fundamental aspects of malaria biology. And today in 2021, we are at a situation where malaria incidence has actually decreased. And in fact, now I am part of what is called as malaria elimination program at the national level. That having decreased the disease burden of malaria, can we actually eliminate malaria from India? And this is really how the science progresses, that you, on the one hand, you identify a problem that's relevant, and then over the years, it's so satisfying to see that the problem actually, maybe in some small and big ways, your own contribution can make some small difference to the disease. Well, of course, in this case, I'm not implying that my science has actually resulted in malaria elimination or malaria decrease in malaria burden, but of course, this was my primary motive when I started working on malaria. But that's an example. And then over the years, I have started, every couple of years, started adding one more disease which is relevant in the country. And I'm going to give you some snapshots. This includes so-called neglected diseases, one rabies being an important one, which is transmitted from dogs to humans. And while all of you know about rabies, it's caused by viruses, a rabies virus, which jumps during dog bite from a dog into humans, and then it causes neurological symptoms, as is shown here, resulting in the death of the human being which has been bit by a rabid dog, and that's it's 100% fatal disease. That means if you contract rabies virus, you're unfortunately, the patient will definitely die. There is no chance of survival. And despite this understanding, despite our awareness of this dreaded disease from prehistoric times, we still do not have a sort of a way of managing this disease. It continues as we speak. Even in the times of COVID, people are actually dying of rabies. Humans are, losing battle against COVID all the time in hospitals, even within Bangalore. This is another area where I've spoken. I'm just going to tell you what we are trying to do here and what is the lacuna here. In rabies, as I said, the main problem actually is that the virus jumps from dogs into humans through dog bite. Now, the challenge really is if there is a timely diagnosis available at the level of the animal, that is already, if you can identify dogs which has contracted rabies, then you would actually quarantine the dog, prevent it from running around, prevent it from biting humans, and thereby the chance of humans contracting the disease is lower. So isolation. So that means first you have to identify the dog, the rabies virus and rabies animal at the source. How do we do it? Second is, once a human actually is bitten and the virus enters, then what? Then of course, as I said, it's only a matter of time that the human is going to die. But during that time, earlier we understand that the human has contracted rabies, better it is for us. Even though we do not have effective medicines, we at least are aware of what's going on. So these are the two aspects of rabies. Now, you will be surprised to know, even in the times of COVID, that the diagnosis of rabies continues to be a major challenge for all of us. Today, there is no 100% method to diagnose rabies. And even more shocking is the fact that the gold standard or the most effective method of diagnosing rabies is only post-mortem. That means only after the animal has died, one takes the biopsy of the brain of the animal and can one confirm by observing negri bodies in the brain that the dog actually had rabies. This is the state of affair with management of disease such as rabies. We do not have. There are some diagnostic techniques available wherein one looks at antibody levels in the blood, looks at the presence of virus by some other approaches, as I will show you. But these are all not 100%. The success rate appears to be only about 15% for these problems. And that's why I think this is why I think we got into understanding that can we develop better methods for rabies infection. Let me just share with you, I think, some of these ideas. I'm going to skip this slide just to highlight that rabies is in fact transmitted through animals and so on. But the most important part that I want to share with you here, and just 1 second, I think I have managed to misplace that slide here, so I'll have to go back. And this is where actually the application of PCR will come in, that how do we actually apply the technologies. As it turns out, one of the most effective ways to diagnose rabies is actually PCR. And that actually the best way to do that is 1 second, please. I think I have misplaced that slide. Yeah, this is what, sorry about that. So the most effective method is actually to be able to detect the virus in the brain sample of the dead animal. But by then it's too late. So what we have done is to develop PCR-based, polymerase chain-based diagnosis using sample from the salivary samples of the potentially infected dogs. Now, this was, funnily enough, even after centuries of presence of rabies virus and so many deaths toll, this method is not commonly available, in fact. And this is what we have done, that taking a small swab from saliva of a dog which is suspected of being infected with rabies, we take it and then perform polymerase chain reaction. The other methods, of course, are rapid fluorescent focus inhibition test, which requires several days in the laboratory, and then direct fluorescent antibody test. These are both antibody-based tests. And then, of course, there is this salivary proteomics, which is something that we are doing currently. And so let me just show you what happens. I think since you are already familiar with the basics of PCR, where one takes the nucleic acid sample, one extracts the nucleic acid and then carries out PCR. So I will not belabor that point again, but what we do is that from the saliva, we ask the veterinary doctor that if you suspect a dog to be suffering from rabies, send us a saliva. The salivary sample we use for extraction of RNA. Now, like COVID-19 or coronavirus, rabies is also an RNA virus. So we extract RNA from the saliva of the rabid dog or potentially suspected rabid dog, and then carry out cDNA synthesis. As all of you know, we have to carry out a reverse transcription reaction using a reverse transcriptase to convert RNA into DNA, and then the DNA is used for PCR by a high fidelity enzyme such as fusion enzyme. This is the World Animal Health Organization recommended primers are used here, and that's really what we do. We have developed our own set of primers, which actually creates even a more sensitive test using the concept of nested PCR. But the main point being that using the salivary sample, we are able to do good old RT-PCR. In this case, of course, these are two-step RT-PCR. In the first step, we are doing reverse transcription to convert RNA to DNA. And then in the second step, the DNA is amplified by PCR and analyzed by the conventional gel electrophoresis. You may have heard about RT-PCR for COVID-19 in this workshop, of course, which is, of course, quantitative RT-PCR, which is basically a probe-based, Tachman probe-based method, which I'm going to come to later. But this is the old-fashioned RT-PCR, where you do a two-step RT-PCR. First, make your reverse transcribe your RNA into DNA, and then the DNA, you add your primers, in this case, rabies virus primers. And what you see here is, of course, the result of such an example there. Negative sample shown in sample 3 does not show you any amplicon. This is your, of course, number 2 is, the positive samples 9 and 10 show you an amplicon at an appropriate size. And also your sample in sample number 2, which is a saliva sample from a potentially rabid dog, shows you the sample, indicating then, of course, that this particular, and then there are other samples here as well, very clearly showing you amplicon corresponding to the.
Excuse me, sir. Sorry to interrupt.
Yeah.
The slide is not shared. Actually, we are seeing the outline of talk slide.
Outline of?
Outline of the talk slide is shared. The actual slide which you're talking about is not shared.
Okay, thanks for pointing that out. I will quickly unshare.
Yeah, thank you, sir.
And go back. Share. Is this okay now? Are you able to see now?
Yes, sir. Now it's fine.
All right, good, good.
Thank you, sir.
Apologies for that. I think what I was showing you here is, the RT-PCR result, wherein you see the negative samples, the 8 sample, lane number 8 is, of course, this now lane numbers have moved a little bit, but this particular lane would be the negative control, where you would see no amplicon, and then positive controls such as last 2 lanes here. These are already diagnosed samples, which we know are positive for rabies, and you see the signals corresponding to rabies, amplicon corresponding to rabies virus. And then the actual sample, which are shown here in this lane, and that lane, again confirming that these are actually positive for rabies. And this is really from the saliva of the suspected dog, then thereby confirming. Of course, this test is very difficult because to be able to extract enough RNA from saliva is not easy, and sometimes it depends on how much viral load there is in the saliva, which disease depends on the stage of the disease. So there is but every time that there is presence of RNA in the saliva or presence of virus in the saliva, you can pick it up. And that's really amazing, because now for the first time, we are able to tell the veterinary doctor or a dog owner that their dog may have contracted rabies. And what that means is, right, that you are able to actually isolate your dog, prevent it from passing on rabies through an accidental scratch or through a bite to either a human or another animal. And that's such a fantastic possibility, because now you are preventing spread of rabies. So what a wonderful tool this is, as you may have seen already, that you are able to, even minute amount of DNA or RNA sample, you can amplify through polymerase chain reaction and then able to detect the presence. So this is, I think, a wonderful example of how PCR, and in this case RT-PCR, can be used for an important application to diagnose infectious diseases such as rabies. And this is something that we commonly do in our laboratory, because we are doing a lot of research on rabies virus. As I said, the other alternative methods which I showed you previously include, of course, immunofluorescence-based method, wherein you would culture the virus in animal cells in the laboratory and look for its infection, or you would do similar direct fluorescent antibody test, which takes, if nothing else, a couple of days. And that's something that is too late, because by then the dog itself may die, or would pass on the disease to other animals and humans. In addition, of course, just to passingly mention that we are now also developing another more effective method of salivary protein-based salivary proteomics by mass spectrometry, which will be even more sensitive than PCR. But just to give you an idea that PCR, about which you have learned so much in the last few days, in fact, becomes very useful for saving lives, just as in case of COVID, also in case of infectious diseases such as rabies virus. What I want to do really in today's presentation is, instead of picking up slides from the net, from Google, for which all of you have access, and if you just do PCR Google, you will get thousands of slides and you can read so much interesting literature. So instead of doing that, what I'm trying to do is to give you some real life examples of how in our own laboratory, in our own initiative out of Indian Institute of Science, we are providing service to the community for applications of PCR, such as rabies virus diagnosis from salivary gland, from saliva of the infected dog. And similarly, we are also able actually to do many other infectious diseases in animals which have the potential to be transmitted to humans. Another example in this case is toxoplasmosis. Toxoplasma infections, which is caused by a protozoan parasite, unlike rabies, which is a virus, toxoplasmosis is transmitted from cats predominantly into humans by various mechanisms. I will not go into the life cycle, but it can go into various animals and then eventually find its way into humans through various possible mechanisms, through food and so on. And in fact, it causes particular threat to pregnant women, because it actually it targets actually the fetus, and thereby there is greater risk for pregnant women. But also it is risky for humans. It can be of course fatal, but it can cause very severe neurological symptoms. That's really how it is. It's not, the fatality rate is not as high as rabies, but it is certainly high, and it causes significant morbidity, particularly neurological symptoms. And so it's a very important neglected infectious disease. And I'll come back to this point in a minute. But even for toxoplasmosis, we have developed a PCR-based solution where we can actually diagnose infection through blood samples of animals and thereby prevent. I'm not going to show you in details of it in too much, but basically just to show you that simple method such as PCR, which actually has so much potential in saving human lives in case of infectious diseases such as the one that I mentioned. The big problem that I'm presenting you today is not that when you think about a technique, it's not just the technique alone that is important, but its application. That's really very important. You have to think about where is the lacunae in the field, where is the lacunae in the community where this particular technique can be of use in increasing our understanding. And this is what, as I've shown you in the slide, that for human infections and animal infections, we have now developed actually PCR-based diagnostics, which in conjunction with doctors, to be able to make people aware about potential threats that humans face through their own infections, but also from animal infections. So if you prevent spread of infections at the level of animals, then you are already in a better shape. That's really the concept through which we did this. And as the title shows, this actually we did on a platform which we developed out of Indian Institute of Science, which is called PET Biotech. And this is what is, I think, would be interesting for the student community here, that as a scientist, you can do that. In addition to doing your research, you can actually reach out to your community through the technologies that you've developed and your clever imagination. In this case, what I've shown you is really an example of that. That through developing platforms of the kind that I showed you, you can actually help community through your science in diagnosing infections such as rabies and toxoplasmosis. But really, these are just two simple examples. There are myriads of other infectious diseases for which diagnostic solutions do not exist, and PCR could be actually one way of diagnosing. But as you'll see in my next few slides, as much as it is easy to understand the principle of PCR, it is, as a science student, it's not difficult to understand the principle of PCR. But to actually use it for a diagnostic purpose is not easy, because you are now, through your polymerase chain reaction study, you are making a claim about whether an infectious agent is present in a clinical sample or not. And so animal's life or a human's life actually depends on the call that you are taking. And that's where the intricacies of this methodologies are, as I will also show you through some of the examples that I'm going to take in case of COVID-19 in the next part of my presentation. I will move on further, and here what I'm going to show you really is not directly about how COVID-19 is diagnosed by RT-PCR, which I think you've already heard from possibly other speakers. But what I want to show you is that as a scientist, how do you actually contribute to the COVID-19 pandemic? My own personal story, wherein the efforts that we have made, how we have tried to engage with the government, how we have tried to engage with ICMR to be able to enhance way of handling COVID-19 pandemic. And it's interesting then that we are still in the middle of it, and everything we talk about in this workshop through other speakers and myself, it's still so relevant for what you are going through. But this is, as Dr. Sushil knows, and many of your professors in your college know, I always like to motivate students, not through technologies, but really through real life stories, real life examples of what one can do, and what, as a young budding scientist, you should do in trying to apply your knowledge of what you are learning in the four walls of your classroom to so many problems that surround us in the society, in the community. That's really what my passion is, to be able to motivate students to apply their knowledge for improving our environment. What that means is not only human, but also for other creatures which surround us. Coming to now this COVID story, I'm quite sure that this slide you have seen several times in this talk, the coronavirus. The interesting story that I'm going to tell you is different from what you may have heard from others. Because my own experiences with COVID-19 have been very unique. I'm showing you this slide really to connect COVID-19 with animal infections. Many of you who are already used to reading phylogenetic trees will have understood, but for those who are not, I will simplify this. The relationship between animal coronaviruses and human coronaviruses. All of us are aware, and believe for most part, that COVID-19 has been transmitted from bats into humans. And then there are the controversies about the lab engineered virus and so on, which I think we will not get into those controversies. But the fact, as I showed you in the earlier slides, that infectious diseases can get transmitted from animals and birds into humans. There have been many other examples, so possibly also COVID-19. But what I'm showing you is the close similarity in COVID-19 genome sequences between the original bat SARS virus and human. The Wuhan with bat, but also the pangolin, which is a kind of an animal, which we don't see here in India so much, but it's very common in Europe and possibly in Americas. That coronavirus that infects pangolin is very similar to the COVID-19 that came out from Wuhan. But then look, it's not too far away. If you look at this phylogenetic tree, to coronaviruses that infect horses, coronaviruses that infect cattle, coronaviruses that infect cats, coronaviruses that infect pigs, and coronaviruses that infect dogs. All of these living beings can be infected by coronaviruses, which are not identical, but are similar to the COVID-19, which has caused this dreaded pandemic. And in case you all don't know already, I will share this with you, that this Wuhan form of COVID-19 is already infecting animals. Wuhan COVID-19 virus is now known to get transmitted from humans into some animals. Luckily for us, the symptoms in animals are not so severe. and it's not always fatal in animals as we understand today. But you may have seen stories in newspapers about COVID-19 being transmitted to lions in zoos, and those are now very confirmed examples. But our own laboratory is doing some extensive analysis of this possibility. But the good news, before you begin to get worried about it, of those of you who actually have animals at home, I would like to tell you that the infection is not so severe in dogs and cats as it is in humans. But the science behind it is what we will talk about today and see how that is actually very relevant. You will be surprised to hear that even before you had heard of COVID-19, already in 2016 and 17, almost 5 years before COVID-19 pandemic happened, my laboratory was actually doing RT-PCR for coronavirus infection in animals, because we knew that this is an infection which is promiscuous, which can infect many species, and it does have the potential to infect humans as well. You can imagine that when I first heard about Wuhan COVID-19, I actually jumped out of my chair, because people are talking about developing an RT-PCR based diagnosis of COVID-19. And here I am, already having a test which I've been using for last several years in animals, in cats and dogs, to study coronavirus infection. So what was such a hard story in 2020 January was something that we already had a grip on very well because of our years of research on coronavirus infections in animals. This slide, right part of the slide, is something that you may have seen several times already in this PCR workshop. So I will not tell you, of course, that what is being done in humans through swabs and RT-PCR, through Tachman probe-based RT-PCR, is something that we were already doing in animals. But of course, there were relatively few takers. But yes, there was a demand of this infection in animals. Now, funnily enough, these infections are called feline. In cats and dogs, they are called feline infectious peritonitis, and then canine respiratory coronaviruses, and so on. And just to, I think, reassure you that these are not so severe for animals, but they do exist, and they are not uncommon in animals for COVID virus. Having said that, I think that I want to now get into a little bit of personal journey of COVID-19. As I already said, that we were actually empowered with this knowledge even before people began aware of COVID-19. In fact, surprisingly, 31st December 2019, when the news came out that pneumonia-like etiology reported in Wuhan, we didn't even know the COVID-19, the name COVID-19 was not yet very clearly given. And then January 7th, when Chinese authorities isolated the virus, and then we heard the name COVID-19, we were actually having a conference in my laboratory through world experts. And we, in fact, we had not gathered to talk about COVID-19, but we actually turned our attention to this early news. There were not many cases reported in India at that time. And experts from Switzerland and other parts of Europe were already in my laboratory talking about COVID-19, and they were pleasantly surprised to know that we already have a test for COVID-19 at a time. In India, RT-PCR was not yet fully developed. And then, of course, we had an early mover's advantage, and over the period of time, we interacted with not only government of Karnataka, but also ICMR to pass on this knowledge and technologies to many other people, including some laboratories in Europe. This is just a snapshot of the conference and a subcommittee that is talking about COVID-19. Some scientists from within Bangalore, and some, of course, visiting from other parts of the world. And that was end of January 2019, remember. And these are now, of course, at that point, of course, government of Karnataka headed, the Minister of Health was Dr. Sudhakar. He right away wrote to ICMR that Professor Utpal Tatu's laboratory already has the technologies and he should be empowered to do the test. And we, of course, moved on, and you would have seen these news articles in newspapers that we were the first actually to create an affordable, low-cost RT-PCR test for COVID-19. And at a time when the price of the test was 4,500 per test, we actually told the ICMR that the price cannot be more than 1,000 rupees because of what we were doing already and what we had done. But that is the fascinating story, that we were at the right place at the right time when government of Karnataka authorities visited my lab together with some industries, because industries were getting closed down, and we were able to make a difference in empowering many laboratories, not only in Karnataka, but this is a letter from an IAS officer from Andhra Pradesh where they requested us to help with our expertise in helping COVID-19 testing in their state. And this is what I want to convey to you, that if you are actually doing something with passion, it actually becomes very relevant in the times of need. And this PCR-based diagnosis of RT-PCR of COVID-19, as I was telling you, the expertise was so common in my laboratory in the last 5 years that we were able to help. And at each stage, this was the COVID consultative group from GOK who thanked us for the efforts that we made to the group which was advising to Chief Minister, and we empowered many labs, because as you can imagine, today you've heard from various diagnostic labs about how they do COVID-19. But at that time, 2 years ago, there were not many trained personnel available. So one of the most important roles that we did as a lab in academia is to start training people for doing RT-PCR, because we were already masters of it. And that's another way by which we did not develop a testing lab ourselves, but we empowered so many testing labs across Karnataka. And as we speak, we are still advising some of the critical aspects of COVID-19 testing through RT-PCR. This is one of the first actually RT-PCR swab being taken in an industry in Bangalore. This was actually already in February and March. This photo was taken on 18th March, but of course this was done by our staff, our personnel, in conjunction with a minister in Karnataka and many other industries. I'm going to cut this long story short, but just to tell you that our interest in helping animal health, our interest in looking at coronavirus infections in animal became so relevant at the time when COVID-19 struck the world, that we were able to pivot and we were able to actually make a difference to not only our own state, Karnataka, not only our own country, but many other countries in terms of empowering. And this was all the simple knowledge of RT-PCR, but more importantly, how do you get robust results which are reliable? How do you avoid false positives? How do you avoid false negatives? As you may have heard, it's a bit complex in terms of calling results of RT-PCR. It's become a commonplace knowledge through newspapers, but this is really what we were able to do with confidence to empower other labs. Now, so those of you who tuned in late, of course, the COVID RT-PCR involves one step ahead of the PCR itself. So through a swab sample where the viruses are present, you make RNA. So RNA extraction, and again here, we did a lot of course, contribution in terms of helping people understand how to get efficient RNA extraction, how to develop standards for RNA extraction, which are acceptable, then carrying out reverse transcription to make cDNA, and then of course, Tachman probe-based RT-PCR to see this exponential rise in the fluorescent signal indicative of the presence of COVID virus. And this threshold value CT, of course, is the main factor, which of course all of you may have heard already from other diagnostic labs, that it gives you an indication of the load of virus, how many virus particles may be there in the sample, depending on the time at which you begin to see exponential rise in the fluorescent signal in your RT-PCR diagnostic method. I will leave some specific questions for the end, but in addition to helping the government in doing this testing, we developed our own kit, in fact. So this was one of the first kits that was developed out of Karnataka, which was approved by ICMR, COVID-19 real-time PCR through Indian Institute of Science in my laboratory, through a platform that we had developed for improving animal health, as I already mentioned in previous slides. But this was one of the first, and now it's being used for COVID testing around the country. I'm going to skip and get into the last part, as I showed you, I'm going to actually, after COVID-19, I'm going to get into the last part of some other applications and exciting applications of PCR, and it'll take me only 5 or 7 minutes. Of course, the application of PCR, or for that matter, any technique that you may learn, is really up to your imagination. You really don't have to Google and find out where can I apply this technology. All you have to do is to use your imagination, that if I have a method of detecting small amount of nucleic acid, where all can we apply it? And this is what I would like the students to remember, that it's really your own motivation and imagination. But of course, these are some of the well-known methods where PCR is used in genetic engineering, diagnosis of genetic diseases, where there are mutations in genes, whether an infection in the environment, genetically modified, all kinds of applications that you'll probably know. But I'm going to give you 2 interesting examples to leave you here. This is a genotyping experiment. Genotyping experiment done in exotic monkeys. And this is something else that we do in our laboratory as a sort of out of interest, that there is these wild animals that we see in forests. Sometimes you can determine their species and their breed through their morphological characteristics. But sometimes you cannot. Morphological characteristics are too subtly different to be able. And then you have to resort to genetic tools. This is an example where a particular species of monkeys, which is called tamarin, we have developed a DNA fingerprint for it for the first time. Up until now, tamarin monkeys were being examined only through morphological features. Some of them are very unique. But now we can actually do a DNA fingerprint. And this is very important for conservation of biodiversity and conservation of species, that you create a DNA fingerprint. And similarly, a closely related monkey called marmoset. We have developed a DNA fingerprint, which again is actually based on PCR reaction. What you are seeing on your screen really is the PCR reaction products of different alleles, which are for this particular animal, and each one giving you a different amplicon size, and that is a fingerprint. That is an indicative of that species. And similarly for marmoset, and as you'll see, the same allele SB2 actually gives a slightly different pattern between tamarin and marmoset. Similarly, I think, for example, for SB7, while tamarin gives you only one single signal, for marmoset, you are seeing multiple signals of amplicons. Each one actually is indicative of this being a different species, in this case marmoset, and so on. In this case, we are talking about species, but also you can do that for breeds, as I will say you. My intention is really to showcase to you potentially exciting applications, and not through just downloading something from Google. But this is done by students like you in my laboratory. And why would we be doing this? Because it's interesting, because it's exciting, and it connects us to the nature. It connects us to applying science to what happens in the environment, in the surrounding, in the life around us. And this is how we were actually doing COVID-19 coronavirus testing in animals. This is how we are doing genotyping in animals. And this is an example of monkeys, but I want to give you, in the last 5 minutes, another example. We all love our birds. We like to see beautiful, colorful birds. But how do they sustain? How do they breed? They can also breed in captivity. There are a lot of bird lovers, and possibly some among you. You may not have realized that sometimes all birds cannot be, the gender cannot be easily differentiated. We are of course familiar with peacocks and peahens, where right away you see that peacock is so morphologically different from peahen. But you'll be surprised to know that many fascinating and beautiful birds, you cannot determine the gender by morphological characteristics. And there, PCR method becomes very handy. You really have to take a piece of the feather of the bird, extract DNA from the feather, and do PCR for a particular marker. Now, the birds, of course, have different sets of chromosomes, as is shown here. Unlike humans, which have XX and XY, birds have ZW and ZZ. So the difference between the male and female is additional Z copy, which is replaced by W, which is a sex chromosome. In this case, I'll not go into the details, but this is how we developed actually a methodology using a particular gene which shows polymorphism between males and females of different species. And using a part of the feather sample, we actually are able to extract DNA from the feather and then able to do polymerase chain reaction using the primers that we have developed, wherein you will begin to see different amplicons. For male, you will see an amplicon at which is slightly different, one at 600, one at 400, whereas for female, you'll see something only at 400 base pairs. So using this simple polymerase chain reaction, something for which you would otherwise have to do laparoscopy. Actually, there are, you have to actually use micro cameras and to go inside the vagina, sort of the sexual organ of the bird to see whether it's a male or a female. Instead of that, through a small piece of the feather, we extract DNA and are able to differentiate whether it's a male or a female bird. And that's very relevant. And that's another exciting application of PCR that we do in our laboratory. And again, any of you who are interested, of course, there are a variety of different amplicons, primers that one can use, and the size, and there's exciting possibilities of how you can even further improve this method of gender determination in animals. A couple of very quickly, I think by similar method you can use for serotyping of bacteria, those which are antibiotic resistant, antibiotic sensitive. An example of that is Klebsiella pneumoniae, which has variety of different serotypes, which actually, all of these are Klebsiella pneumoniae, but the serotypes are different. And that then, again, by looking at certain microsatellite markers, you can develop primers and do PCR. And here's one example that all these different serotypes can be differentiated based on the presence or absence of a particular primer, a particular amplicon corresponding to specific to a particular K2 serotype of Klebsiella versus K1. And the reason it's important to do serotyping is because they exhibit different antibiotic sensitivities, that certain serotypes exhibit antibiotics resistance, and thereby if you already knew, just knowing Klebsiella pneumoniae is the causative agent is not enough. If you knew the serotype, you would actually avoid giving certain antibiotics and give others which are working against drug-resistant form of Klebsiella pneumoniae. So that's another application that we do in our laboratory. And finally, something very exciting and something that will appeal to the young people, that horses. Horses are so beautiful animals. In Bangalore, you see Bangalore Turf Club and so many beautiful animals racing, race horses. There are a variety of species of horses, a variety of breeds. One of the interesting ways by which we have applied the method of PCR is to characterize one of the most interesting and oldest Indian breed of horses that are called Marwari horses. Those were, of course, native to Indian subcontinent, but over the years, their importance has gone down. But as a scientist, I was very passionate that I want to study and preserve what was native to our country. And so we developed methods wherein we developed DNA fingerprint of native Marwari breed of India so that we can actually identify and confirm that this particular breed is in fact a Marwari breed. And not only that, we are now using this technology of PCR-based genotyping to actually help owners of such horses to confirm and then accordingly breed pairs which are Marwaris, and thereby propagate this limited gene pool that is still available in our country before it becomes extinct. Because these horses are actually a mirror of our Indian history, our culture in many different ways. So this is really where, again, the technology is the same. You extract genomic DNA, in this case from the hair of the dog, or you can also do it from the blood sample. But why take a blood sample when you can just take a piece of hair and extract DNA? And this is what we do. And we have developed certain microsatellite markers to do this. And then we carry out polymerase chain reaction. This is again done by students like you, and you can actually resolve. There are several alleles which showing amplicons of different sizes for different primer sets, which are characteristically present and just to tell you that these gels are not agarose gels. These are low percent polyacrylamide gels because these amplicon sizes are below 500 base pairs. And so you need slightly different gel systems for differentiating it. But just another example of, and then we provide this kind of a data of not only the morphological characteristics, but also the allelic frequencies based on the PCR reaction that confirm, and to what extent, not just presence or whether or not it's Marwari, but how close it is to the original Marwari breed is what we are able to give by these methods. So with this, I think I'm coming to end of my time, and I'd like to leave a few minutes if possible. But I want to thank again Dr. Sushil and all the faculty that they have done such a wonderful job to expose our students to this important topic. My own motivation is that we will get some bright students to work with us, because I have always enjoyed working with students from Ammanni College. So I would welcome any student who has excited about applying science to such aspects of animal health, animal well-being, human health. We always have space for motivated people. With that, thank you so very much for your kind and patient hearing.
Thank you very much, Professor Tatu. It was great talk. I am always mesmerizing with your whatever you speak about. It's like great listening to you always, as usual. So now session is open for the questions. So let's hear some questions. We can take few questions. So let me see the chat box. Can medicinal plant from India that have been stolen by other countries be confirmed to be Indian origin using PCR? The
Absolutely.
question is, yeah.
Yes. I think, yes, the simple answer would be yes. So this would be definitely an application. But, again, how the important thing is to prove that these were originally in India. That is the important part, actually. So you can claim a species or a particular variety of plant to be identification by PCR. For sure, you can do it. But now, how are you going to prove that that was originally from India is another question. But yes, I understand your emotions behind it, and one can use PCR for identification. Yes.
Yeah. Thank you, sir. So next question says, what was your objective to initiate and do research on coronaviruses in 2015? Way back it is.
That's why I think you missed that. Coronavirus infections are common in animals. They did not start in 2020 or 2019. Animals, and in fact also humans, coronavirus infections have happened even before that for a long time. It's just that COVID-19 is a more severe form, and that's why so we were actually looking at coronavirus infections in animals for last several years, because it's one of the most common infections in animals, and it causes a lot of morbidity and sometimes fatality. And that is why, and that's really what the main point I wanted to convey to you, that as a researcher, if you pay attention to problems which are not yet addressed, and that is what we did. We were focusing on coronavirus even before it became a pandemic, at the source, at the level of animal. And as you can appreciate, in fact, it has jumped from bat in this case. But we were not looking at bats. We were looking at cats and dogs and horses and cattle, all of which are susceptible to coronavirus infections. That was really why we wanted to improve animal health. And also for the reason that we were kind of aware in our scientific thinking that just like influenza virus has jumped from birds into humans and from pigs into humans, there is a possibility that coronavirus, we had that thinking already in 2015. So that's the answer to your question. And that's the most exciting part of what we have done in the last several years.
Yeah, it's too exciting listening the questions and answer. Another question says, the vaccine, what we are taking now, the same vaccine can be administered into the animals?
Very interesting question. I think we'll have to have another seminar, but I will tell you a secret. Even before COVID-19 happened, there was already a vaccine for coronavirus infections in animals.
True.
Okay? So and which we were actually giving after the, in animals which actually were susceptible to coronavirus infection. So coronavirus vaccine for animals existed even before COVID-19. And I can talk more about it, but I think this is there are so many things that we don't know. It's amazing the way the science progresses. But anyway, that's the answer to your question. Very good question.
Yeah, thank you, sir. Is there any possibility to have false negative results using PCR?
Absolutely. There's always a possibility, and as you see in newspapers all the time, people who have COVID symptoms sometimes don't test positive, and there are all kinds of complications. One is because of the technical challenges. Sometimes people, there can be errors made by humans. That's possible. You cannot rule it out. But also sometimes the viral loads are low at a particular stage. And so there are various possibilities why somebody who's actually COVID positive may test negative by RT-PCR because of some technical challenges. And there are many technical challenges. And you can imagine that when labs are testing thousands and thousands of samples per day, imagine 1.3 crore people in a country, how much testing can anybody do? And during that time, sometimes technical challenges do crop up, but that could be one reason why you can have false negatives, but also sometimes because of genuine reason that the virus count is not high enough to be detected even by RT-PCR. It's very low virus count. So such cases you can have false negatives.
Another question says, sir, what was the actual reason for rapid spread of virus in recent years, though virus was present in early years?
Absolutely. I think this is an exciting possible. What has happened is that like in case of influenza, influenza was also there, and then suddenly it came again because it underwent mutations. So also coronavirus, which was infecting bats, which was present in bats, underwent some mutations that made it possible to now aggressively infect humans and spread between animals and humans. It's the mutations in the spike glycoprotein predominantly that drove evolution of this aggressive form of COVID-19. That's the simple answer, but it's a much more complex story than that. But that answer should satisfy you at the moment, yes.
Another question says, can dogs or cats spread them to human?
It doesn't appear so far, actually. But nature is so complex. So I think as a scientist, we know that animals can transmit infections to humans, as I gave example of rabies or some other infections, toxoplasmosis, and that's really why I gave those examples. So it's not unthinkable. We do know that animals can transmit infections to humans, which are known infections, but in case of COVID-19, we don't know that enough. It appears it's a lacunae currently in our thinking. It doesn't appear that currently we have that problem, but it's an interesting question, and I think how the nature's progression plays out, only time can tell, I think. Yes. But yes, so far we don't have that threat.
Truly said, sir. So another question says, it's very basic question, I feel so. How long do COVID antibodies last in the body? And why is there a gap of 1 to 2 months between the vaccination doses?
Yeah, I think the gap in 1 or 2 months is only in this particular vaccine, AstraZeneca, the Covishield, because of some complexities of the way the vectors are used, adenoviral vectors. And that's how they found out that the longer time you give for the second dose, the better chance of boost of the immunity. That's really the case for Covishield, but that's not true for many other vaccines. For example, Covaxin does not require such a long gap, as you already know. And other Moderna and all those also probably don't claim to have such big gaps. Sputnik doesn't have such big gaps. And now we are even looking at single shot vaccines that are going to come out in the market. So yes, but for Covishield, you are right. The gap is big, and there's a technical reason for it because of the way the adenoviral vectors are used for as a vector to host the COVID spike glycoprotein in this case. So there are some technical, yeah, go ahead.
It's like a booster dose. Yeah. So next question says, if according to research, COVID-19 lab developed or natural mutation occurred. So it's just want to say whether it is nature made or it's lab made. The question says.
Yeah, it's a bit of there is still a lack of clarity on it. And I do not want to get into controversies by making statements. All I would say is that this is not the first time that an infection has come from animals or birds into humans. This is not the first time that an infection has come from bats into humans. Middle East respiratory syndrome virus, MERS, original SARS-CoV also came from bat. These are both examples. One of them came from camel, other came from. So coronavirus infections have come from bats in the past. So I do not find it challenging to accept that it could have come from bats. But I'm not against scientific evidence. So I do not want to get into controversial statements, but I'm more focused on the science that yes, infections can come from animals into humans. And that's something we should be aware of, not just in case of COVID-19. It's not going to be the last infection. There will be many more in the future, unfortunately.
Sir, just to continue with, thank you very much for your enlightening. So just to continue with, in 2012, if you remember this, is when they drawn a phylogenetic tree, it was equally managing with that. It's 90% with R1, RX1, so that other viruses. So is that true? It has come from that lab itself?
I think it's very hard for me to comment on such controversial topics, Dr. Sushil. So I would avoid commenting on these, and I would rather focus just on the science and scientific facts. And so I apologize for not being able to address this.
It's okay, sir. So what is the difference between Covaxin and Covishield?
Well, both of Covaxin, of course, is an inactivated whole virus. So it's a virus which is inactivated so that it cannot carry out infection, and it has all the components of the virus. And thereby, our immunity will react against multiple aspects of the virus, multiple proteins of the virus. Whereas Covishield is merely spike glycoprotein, which is with the DNA for spike glycoprotein, so only one protein. So main difference is that Covaxin has multiple proteins, Covishield has only one, and it has DNA. It's a DNA virus, Covishield, whereas Covaxin is actually a protein virus, a protein vaccine.
What about the cocktail of giving them?
It's possible. I think now people are thinking about first dose with one vaccine, second dose with other vaccine, and all of these possibilities are now cropping up to find what is the most optimal, what will be the most effective way to confer immunity.
Sir, is it possible someone to take the booster dose in India after taking Pfizer vaccine?
Yeah, it's possible. Absolutely. I think yes, there are people who would take first Pfizer and then come to India and take Covishield here. Yes, it's possible. Absolutely.
What about the symptoms? Is it same as we have in human about the corona?
In animals?
Yes, sir. With respect to corona.
They can be similar, but they can be, sometimes animals have coronavirus infections causing variety of different symptoms, including some cases they cause gastroenteritis. Sometimes they have respiratory problem like humans, but variety of other possibilities. Actually, they manifest in various different ways because there are so many forms of coronaviruses. There are so many different kinds of coronaviruses, each causing a different kind of infection. And I give you, in fact, example of feline infectious peritonitis in cats, which is one very common infection in cats. But yes, it can cause different symptoms than in humans.
Very interesting, sir. So then, can using efficient proofreading reduce the percentage of false negative in the diagnostic of not only COVID-19, but other diseases also?
Yes, absolutely. I think there's always a possibility to improve the sensitivity and specificity, as we call it, for using improved methodologies. There are constant improvements happening in the enzymes, in the protocols. So absolutely, this is an ongoing story. And as newer and newer kits are coming out, they offer better and better specificities and sensitivities for diagnosis. Yes.
Is there a possibility PCR can be used for understanding scam, Ayurvedic medicines, complementary alternative medicines?
Absolutely, why not? Because if you have to determine a particular plant species, you could actually use it for that purpose. And so you can create a barcode for DNA barcode for a particular decoction of the Ayurveda medicine. Yes, it's possible.
Can we get to know the amount of viral load in RT-PCR?
Yes, you can. Because that's where I was talking about the CT values, so CQ values. That is an indicator of the viral load. So you can actually quantitate virus, and that's why it's often called as a quantitative PCR. You can quantitate the load of nucleic acid by RT-PCR.
Yeah, it is another question says, any possibilities to get affected coronaviruses without any symptoms?
Yeah, absolutely. I think we hear about asymptomatic patients all the time. So it is true that there are many of us would have had coronavirus or even COVID-19 infections without showing symptoms. It's absolutely possible.
True, sir. That is truly known.
Yes.
And just look for another questions. Sir, will this pandemic truly be over? When will the pandemic truly be over?
I think that's a very important question, which I don't think anybody in this world has the answer to. But we do know that now the vaccination kicking in, we are going to see an end to it. It's not going to be endless. We will have significant immunity as more the populations are getting vaccinated, and that's going to decrease the load of. But there will be waves, small and big waves that will happen. The big challenge is how do you vaccinate the vast population in our country, which is, today we have barely less than 10% of the population vaccinated. We still have vast majority of our people not vaccinated. And that's really the big challenge. But if you assume in many other countries where vaccination has, because of the smaller populations in those countries, has become, most of the population is already vaccinated. And there is the peaks are going to be low. The virus, the pandemic is going to be lower in intensity. And that's why those countries have opened up a bit more. But then there's always this threat of a major mutation that may challenge the vaccine. But I don't think there'll be a big problem. I'm optimistic and think that in the next few years, we are going to see gradual decrease in the load of COVID-19 all over the world. But it'll be a slow and steady process. It's not going to be sudden. It's going to take a few years.
Truly said, sir. Thank you very much, sir. Sir, with the advent of microfluid PCR technique and the CRISPR-Cas, can genetic disorder be corrected? If so, can those disorders that are currently deemed incurable, like neurodegenerated, can be cured?
Yes, there is a hope that gene therapy through this newly developed CRISPR-Cas technologies are showing promise to correct genetic disorders. And yes, I think there's already a lot happening, many examples where there are successful experimental level successes, which now are moving towards field applications. But it does hold. But there's this it's a double-edged sword, because CRISPR-Cas can also cause damage in some ways. I think it also is if it goes in the hands of wrong people, it can become a harmful tool. So that's where I think there are a lot of ethical issues associated with use of CRISPR-Cas in manipulating genomes, and so on. But yes, in principle, as far as the science is concerned, it has the potential to correct genetic disorders.
Sir, what is the symptoms for the Delta plus variants?
Symptoms are not that different. It's just that I think Delta plus has the ability, as we understand, to spread faster because it's able to actually move. It has a greater transmissibility because of certain mutations in the spike glycoprotein that makes it, on the one hand, infect faster. Also, on the other hand, the fusogenic capacity is higher of its membrane fusion protein, spike glycoprotein fusion capacity is higher. So yes, it is. That's the reason it is a matter of concern.
Why it is more effective in children?
We don't know that for sure yet, actually, whether it is more effective in children, but that's some data that suggests that. But we don't fully, I don't think there is scientific evidence yet to say that it is more effective for children.
Okay. Why the rapid testing for COVID less efficient in comparison to RT-PCR?
That's because it's antigen-based testing, and rapid antigen test has a limitation because the amount of virus required to get positive is much higher. So your sample should have very high viral load to be tested positive by rapid antigen test, as compared to RT-PCR, which even minute amounts, much lower amounts can be detected.
Yeah, I feel there are no much questions now. So thank you very much, sir. So it was a great listening to you as usual. So the if any other questions are there, you can always send a mail to sir tatu@isc.ac.in.
I have a question. Like
Please.
the sample and the methodology which you have used in 2015, is it the same which is introduced now for the diagnosis of SARS-CoV?
No, see, in animals, it can cause various kinds of symptoms. So I think it can. We used to use blood sample quite a lot. Sometimes exudate, because there are various forms of coronavirus infections in animals. So it depends on what kind of symptoms and what kind of disease it was, and depend on that. But typically, one uses the exudates, which are accumulation of fluid in the body, peritoneal fluid. That often is, but also sometimes blood sample. Sometimes viremia is high. You can detect it in blood. So different kinds of samples.
Last but not the least, sir, like the coronavirus was been discovered in 1960. Since then, there are many progress which has been done in the field of vaccine. So, do you think that the vaccines which we have seen now, is there any chances that they are going to develop the resistance for those vaccine as well?
It's not unthinkable, but I think given that we have such a spectrum of different vaccines available, some which are whole virus based, some against certain proteins and so on, I think we will be better, we'll be in good shape to counter the new mutants that may appear in future. I do think that we will have novel vaccines in our armory to even take care of mutants. Actually, as it appears, the current set of vaccines are actually working against many of the new mutations which came after the vaccines were developed. So I'm very optimistic about it.
Thank you so much for answering the question, sir.
So thank you very much, sir, for answering the questions. Very interesting way. I think the audience would have been clear with their doubts, and they all will be having. They will all will be clear with now, and they will be having now new objectives in their lives. They will be how to understand the science and how to see in a different perspectives, as usually you do. So just, sir, I want to ask you one thing. Every 4 years, you said you are going to you changing the objectives of your research. From where you get so many ideas?
Well, I'm not changing the objectives of my research, Dr. Sushil. I'm just adding new important topics. So objectives are the same, of course, but we are adding new questions, new problems, because as also, as you can imagine, the problems in our environment, in our country, are endless. Our country has so many problems. In terms of scientific problems, infectious disease burden, and there are so many very basic infectious diseases not being studied enough, and that's why we call them neglected. Simple things like diarrhea. Even today, in the middle of COVID-19 pandemic, it's a fact that children below the age of 5 in many parts of India die because of simple diarrhea. So I think, so it's really not hard to find important problems in our country or anywhere in the world to be able to, try and apply your science to it. So that's really what my effort has been, that to be able to find questions where there are not enough scientific interventions available, and then try and make a small difference to an extent that we can.
So thank you very much, sir. There are no more questions out
Sorry
there.
to interrupt.
If anyone can, yeah, please.
Yeah, sir. So my question is for the professor. So since you told you're very encouraging for the students who are really interested in things like this, I just wanted to ask if you offer any internship programs or any hand-in experiences for all this RT-PCR and PCR techniques. Because, listening to all this online, it obviously has built up too much curiosity to get in a hand-in experience and do a laboratory way of learning.
Right. No, absolutely. As I said, we always welcome interns and people who want to work with us. But, we also have, the way I would look at it is, we we also the limitation we have is the number of people in the lab. So we can't run a training program constantly because. But on the other hand, I think the best way to learn is to actually participate in our programs. And that's where I always welcome students from Ammanni, that you can come and work as interns and also continue to work with us for a little bit. That's when you really get the flavor of it. It's not just that you run a couple of times a PCR reaction. That you can probably, I'm sure you can do that in your own college. But if you want to participate in a research effort, then I think these are absolutely, we welcome young people to come and work with us, because it's not just about the techniques, it's about the enthusiasm of being able to solve scientific problems and solve problems in the community. So people who have enthusiasm for it, we are always welcomed in our laboratory. And yes, it will be a delight to have students come and work with us.
Thank you, sir. Thank you so much. That means a lot. Please share the information for the same. Getting
Excuse me,
Madam,
sir.
you
I
can
have a
send
question.
like 1 minute, ma'am. Well, you can mail your information to the tatu@isc.ac.in. Other questions, whatever question, we are already running short of time. So you can always go through with the sir's web page on ISC. All information is available. Even if you want, you can mail him to tatu@isc.ac.in. He will politely answers you always. I know that he always takes up the questions. He always reply you back and as soon as possible. So we are already running short of time because the next speaker and next quiz competition is going to start. So I feel so. So we cannot take more questions now. If anything is there, you can always go for tatu@isc.ac.in or write to us. We will forward to sir. So thank you. So I'm sorry to stopping everyone here. I know there are a lot of curiosity, lot of questions, and because the way sir explains, I know that there are a lot of questions arises, and he is very clear in everything. So he's a pender of knowledge. I can understand. So please uh, believe me, we like timing is the only constraint here. So if you have any questions, please write to us. We will go for it. Okay. So now we have to close the session because of time limit. So thank you very much, ma'am. Thank you very much, madam, for asking the question. Sanjana, madam, and whatever sirs and students, all participants asking for the questions. So I have to thank now Dr. Utpal sir. Thank you very much, sir. It was delightfully and very simply taught and very easy to understand the concept the way you have taught us. You have told us as usual. I am always delighted to listen you at every workshop, every conferences. So thank you very much. And you are right. You rightly said you are a great mentor to all of us. For me, at least. So thank you very much for sir, for accepting
Thank you.
our invitation. Thank you very much, sir.
Thank you very
So
much. Thank you.
thank you very much. Thank you to all the participants. Thank you, sir. Eva, madam, you can take over.
Sure.
Take over, please.
Yeah, sure. I will just make an announcement now. See, participants, since we have, run half an hour ahead of the schedule. Okay, so I will do one thing. Take a 5 minutes break. Okay, and go back to the Google Classroom and start attempting the quiz competition. I'll give a 5 minutes break for you now. Okay, another 5 minutes. I will post the Google form in the Google Classroom. You can join and you can attempt. Is it clear?
Yes, ma'am.
Yes. Take 5 minutes break and join. Yes. Okay. Yeah. Okay, sir. Thank you.
Thank you, ma'am. So let's sign out. Happy learning. See you soon.
Good afternoon, all. We'll just start in few more minutes. The afternoon session.
Ranjita, Yashaswini has joined.
Yeah, she's there.
Yeah, ma'am, I have jumped.
Okay, okay.
Shall we start, Hema?
Ma'am, how many participants are there, ma'am?
75 are there, Hema.
We'll wait for another 2-3 minutes, ma'am.
Okay, fine.
Yashaswini, is it off for you today?
No, I'm at company itself.
You're there in the office itself. Okay.
Yeah,
How
ma'am,
is the
I've
number of
taken 1 hour
samples that
permission.
you're getting today?
It
Nowadays,
has
how
been
many?
reduced now, ma'am.
It is reduced.
Around 500 sample. Yeah.
That only is reduced. Otherwise?
Yeah, ma'am. Usually during the second wave, that is during March and April, it was 1,000 to 1,500 sample.
Oh, okay.
We used to get 1,000 to 1,500. Now it has been reduced to 500.
And what is your staff strength? How much each of you get to process?
Hello?
Can you hear?
I'm not able to, no, not so clearly, ma'am.
Okay. So how many of you are there staff strength, and how much does each one of you get to process the samples?
Actually, ma'am, when we used to get around 1,500 samples and all, now we have completely automated extractors. So
Oh, okay.
per person can do 100 sample per hour, around hour. So one person can do around 300. If it is manual extraction, one person can do around 50 for 2 hours.
Oh, okay. So it's been how many months that you are there?
It's been 10 months now, ma'am.
Oh, good.
Hello, Yashaswini.
Yes, ma'am.
Yes, how are you, Yashaswini?
I'm good, ma'am. How are you, ma'am?
Fine, dear. So nice to see you that you are associating in our workshop.
Thank you, ma'am. Thank you for the great opportunity.
No, still I think you can explore much more. I was just listening to the discussion with Hema ma'am. So
Yes, ma'am.
even though you are a young budding entrepreneur, but still you can always go ahead.
Yes, ma'am.
So you are at workplace, is it?
Yeah, ma'am. I'm at workplace itself. I've taken permission for presentation.
Okay. So nice to see you, Ma. And that too at this junction, no, I think even we need to learn much more from you.
Thank you, ma'am.
Okay, all the best wishes.
Thank you, ma'am.
Babita and Hema, shall we proceed? It's 102 participants in.
Yes, ma'am, we will start. Yeah, we'll start.
Good afternoon, one and all present here. I am Dr. Gauri Nilima from Biotechnology Department. here to introduce our young budding enthusiastic past student, Yashaswini here, so that she is one of the very enthusiastic from her studies during her bachelor's only. We have seen her increasing her enthusiasm, and after her PG also her enthusiasm to learn more into the same uh, arena of research made her brought back here to explain you more about her research experiences. She has learnt a lot about different types of nucleic acid extractions, RT-PCR analysis, real-time PCRs, and she has learnt lots of culturing techniques and use of semi-automated or antibiotic sensitivity tests and all. And he is also one among her colleagues who has been selected for training for cytogenetics for a national reference lab. So she has been testing CV and COVID testing right now from past 10 to 12 months. And now she is on for more on to molecular diagnostics related to oncology tests. Not only this, to say to her credit, she has also attended lots of workshops, seminars, and she has done her projects under UG as well as a PG. And to say, we have been very proud of her that she won the Best Poster Award of the year 2018-19 for the project Production of Lipase by Solid Waste Fermentation of Biofuel Seed Cake and its use as a biocatalyst in biodiesel production. This was awarded to her by KSCST at 42nd series of student project program. So a round of applause to Yashaswini. Thank you for being with us, and hope all of you will enjoy her talk in a more enthusiastic way. Over to you, Yashaswini.
Thank you, ma'am. Thank you for great opportunity you teachers have given me to present here. This is the first opportunity what I've got. Thank you very much for all of you, ma'am.
Thank you very much. Surely we'll have great days ahead.
Yes, ma'am. Hello, everyone present here. Good afternoon. I'm here to present you about COVID-19 diagnosis using RT-PCR. As you all know, now COVID-19 diagnosis, especially RT-PCR, has been a limelight in molecular diagnostics department now. It's so important that everywhere you get to hear RT-PCR and RT-PCR. So it's very important, one of the important technique right now. Before starting, I would like to ask one simple question. Why are we using RT-PCR itself? Why not normal conventional PCR? It's my question to you, all of you.
Because coronavirus is an RNA virus.
Okay.
Like you
And
need to
then,
convert the sample which is there, which is a viral, which has the genetic material, that is RNA, should be converted into DNA back. So according to the central dogma of molecular biology, a conversion of DNA into RNA back, it is called as reverse transcription process.
Okay, you are right. But I would say cDNA can be done without using RT-PCR also. We can synthesize cDNA without RT-PCR too. Why we require RT-PCR then?
For expression studies, like cDNA does not contain any since it's an RNA virus, if it's directly if RT-PCR is done, like only the translating regions are expressed, only the expressing regions, the exons. So the disease can be detected.
Yeah, but main thing I will say is, first of all, if you have to go, ma'am, can you put for slide 1? Hello. Thank you. So if we go for just imagine if we go for traditional PCR, we would be isolating the RNA and we would go for cDNA synthesis separately and then do the PCR amplification in a general conventional PCR. And then if we have to analyze, then again we have to go into agarose gel electrophoresis, which is again the tedious process. And if you observe, going for all these steps separately, it would increase the chances of contamination too, which will give the false results. Analysis of data also becomes tedious and process becomes very tedious. So whatever RT-PCR we are doing now, it's a one-step PCR where we'll have cDNA synthesis happening inside, PCR amplification happening inside, and also along with measurement of the product with fluorescence. All together, it is happening in one single step. It becomes so easy and it decreases the contamination also. That is why we are using RT-PCR. On slide 2. What is happening inside the RT-PCR is initially RNA extracted is loaded into the machine with enzymes which is required to convert it into cDNA. So initially, first step is around 95 degrees Celsius temperature, cDNA synthesis occurs. And after cDNA synthesis, this is denatured. It is denatured around again 95 degrees Celsius, and it is denatured, and then reverse primer and forward primers. There is forward and reverse primers comes and binds into it. This is annealing step. During annealing step, there is one more thing happening here. Along with forward and reverse primer, there is something called Tachman probes which comes and binds to it, which is having the fluorophore molecule in it. So when light is excited, this cyclus has a light source which excites onto the, that light source will excite on this fluorophore molecule, and this will emit the fluorescence light. So this fluorescence light is captured as a digital data in the software, and this we do the analysis of the data. This is how it works inside. And at last, extension happens, and you would ask me, this Tachman probes would have been bound in between, wherever sequence complementarity is found. So if that has to be removed, obviously DNA polymerase comes into picture, where DNA polymerase is having 5' polymerase, 5' exonuclease activity. So this digests the Tachman probes, and these fluorophore molecules are released. So which get excited and releases the fluorescence. So that is that gets captured in the system. That is how we do the analysis of the data. Next slide. And initially, if we have to start with the process, sample collection is one of the critical step, we can say. So usually sample collected are upper respiratory specimen samples like nasopharyngeal, oropharyngeal samples. So you would ask me why this lowest, I would say in upper respiratory specimen samples, like they consider only for patients who is recently infected or recently seen with onset of symptoms, like 5 days of virus incubation period. So after that, usually what they does is they will not collect if patient is severe, having the severity of the disease, and mostly it is infected the lungs. It has been almost many days after onset of infection. Then they would not go for upper respiratory tract because the sample would not be found in the throat or the oropharyngeal or nasopharyngeal. They would go for lower respiratory specimens or else sputum if they produce. So this is how samples are collected, and usually these are collected in VTM vials. VTM vials is viral transport media, and the speciality of this viral transport media is this is containing antifungal and antibiotics in it, so that it doesn't allow any bacteria and fungus to grow in it, so that it doesn't interfere with nucleic acid of what we are targeting for, that is COVID nucleic acids. Apart from that, it doesn't contaminate the sample, I mean to say. And then this sample, what we receive, usually they send it in ice packs. That is, they maintain the temperature of 2 to 8 degrees Celsius so that it doesn't degrade or less. The loss of stability. Stability is not lost in the sample, so that they maintain in 2 to 8 degrees Celsius while transporting it into the lab. If there is any delay in processing, then we can maintain that in -22, so that we can store it and we can process it later. And these samples, not only, I would like to tell, while decontamination, not only receiving the sample is important, the decontamination procedure of the sample is also very important here in the labs, so that our environment is not affected. So usually we do autoclaving in the lab itself. And apart from that, when government takes a sample, they do incineration also. Next slide. So this is how it comes. Like usually whatever that picture it is there, above the samples also they would have kept ice. Below the sample also they would have kept ice. It comes in thermocol box. These are the VTM vials. And we'll go for first step, that is RNA extraction. So before that, I would like to tell you how these lab area will be divided in molecular diagnostics. So initially, before we go to RNA extraction, where we are handling pathogenic samples, we are supposed to cover ourselves. That is where PPE comes into picture, personal protective equipment. So we have two areas, which is called donning area. One is donning on and one is donning off. So donning on is a place we wear the PPE and get into the extraction room. And in molecular diagnostics, it is always a single way, where we get into the extraction room and we get into the extraction room and come out in the same way. It is always a single route way. So we wear the PPE and go into extraction room. And usually in all of these pathogenic handling RNA nucleic acid extractions room, they are well equipped with biosafety cabinet, and also room is also equipped with HEPA filters. It is actually called pressure rooms, like negative pressure. Usually in extraction rooms, they maintain negative pressure. That is because whatever the contaminated air is present inside the room, it should not go out of the room. So what that does is, the pressure inside the extraction room is maintained in such a way that it is different when compared to the pressure outside of the room. So whatever contaminants like organisms, it cannot go out of the room. And then along with that, those negative pressure also has HEPA filters, which filters the air inside, so that technician whosoever present inside, he will also be in good condition. The air inside also it will be in good condition. And by the way, if by any chance the air is also penetrating outside, it is filtered and then left to the environment. So it doesn't affect the technician working there, and also the environment. This is how extraction departments, extraction areas will be maintained. And then after that, we go to donning off area, where we remove our PP, and obviously our PPs are single wear. We dispose it at single time. And then after that, next area comes to master mix preparation. Master mix preparation, it is a completely clean room, because even if there is a little bit contamination in that room, it might affect our results completely. So that is a completely clean room. And after that, next area comes for template addition room. We have master mix is separate. There only the master mix reagent will be prepared. And in template addition room, whatever RNA extraction room is there, actually RNA extraction room, this template addition room will be connected in a single piece of door, just like a window, where this template, whatever RNA elutes we have got, that one we will not be carrying it in hand and will not be moving it around, because that can even contaminate other areas. So always we have a connection to template addition room and RNA extraction room, where they just pass it through that window kind of into the template addition room. So we add the RNA template there into the master mix plate, where already prepared. And after the template addition, there is a separate area for positive control addition. That is also one separate room where controls are added. And apart from that, at last area is this RT-PCR setup and analysis room. So this is how it is. And now we'll go to RNA extraction. So there are two methods in RNA extraction. One is manual extraction and one is automated extraction. I will tell about manual extraction first. So this is basically based on, it is completely on pipetting and it has many steps. So whatever steps machine does, that all steps will be doing it, and is completely based on spin column based. Someone is asking question. Ma'am, can I answer it at the last or should I answer it now?
Aishwani, we can take up the questions after your talk.
Okay.
I think at the end of the session, we can address them.
Okay. So
Yes, please continue.
Yes, ma'am. So in manual extraction is completely based on spin columns. These are the spin columns you can see there. Those columns are actually made up of silica. So due to charges and salt concentration, what happens is RNA can bind to this silica due to charges mainly. So usually what we does, can you go to the next slide? So this is a common steps for extraction of RNA. So basically the tube what we have will add the sample with lysis buffer. So lysis buffer, main principle of this lysis buffer is to lyse the protein envelope of the virus. So it will lyse the protein and let the nucleic acids out into the solution. So basically, initially we add the lysis buffer, and for different protocols, for different viruses, they have a different temperature, room temperature incubation, hot water bath incubation, dry water bath incubation, like that. Many protocols are there. So usually in this COVID, we do the room temperature incubation itself. And then after incubation, we go for centrifugation. So as I said, this is spin column based method. It's completely vacuum based, where centrifugation is done in all of the steps. So after that, we would like what happens, this RNA, whatever nucleic acid has come out, it would have bound to the silica. So what we have to do now, whatever extra things is there, like for example, proteins, it has been ruptured, but we don't want that. We have to remove that. So next step, we go for wash buffer. We add wash buffers. There'll be 1-step wash buffer, 2-step wash buffer. Like that, it depends on the protocol again. So we add wash buffers to remove the contaminants again. And at last, the RNA is still in the silica. We want it down in the elution tube or collection tube. So we add elution buffer. Here basically it is opposite of lysis buffer. In lysis buffer, it is having high salt concentration. So instead of water bound to the silica, this RNA goes and binds to the silica. And here in elution buffer, what happens? Again now water will go and bind back again. RNA will come down in the elution with elution buffer. Due to pH or salt based, it will elute out into the collection tube. So in lysis buffer, mainly here it is guanidine salts which is present in the lysis buffer, which is highly denaturating. And in wash and all of these, actually, if we are getting the commercial kits, usually that's what happens here. They procure us the commercial kits. That doesn't mention anything. It's just mentioned as lysis buffer. It's just mentioned as wash 1, wash 2 buffer and elution buffer. We have to go into it and study something about it. It's how like all commercial kits are. And this is a place of extraction in biosafety cabinet. This is template addition room again. For this, we use laminar airflow itself, template addition room. Now we come into important step of quality control. For all the steps to ensure that whatever we are doing is correct, we require quality. So for example, in extraction, we would observe, for example, if we receive a sample from a patient who is like incubation period of virus is less than 5 days. just imagine if it's less than 5 days and there is a not enough amount of virus which can be detected in the machine. So if it's in such a case, what happens? Like we get a false negative. Like for example, that can also happen. Or else even if we does the extraction to show whether we have done extraction properly, we require controls there also. So in every step we have a quality control. For example, if we go to extraction, we take a RNase free water and extract it along with our samples. So obviously RNase free water doesn't contain any nucleic acids. So what should happen? No amplification should occur in that when we run it in the machine. So that is one of the control we have. Like that in master mix, I told you there is separate room for positive control. So whatever genes we are targeting, there is a positive control which is positive for all the genes. Like it is completely positive elute kind of, which is having all the targets, it is present. So that we'll use as a positive control, and usually negative and positive control will be used to compare the curves of patient sample also. And one more thing is there which is very important. To eliminate false negative results, we use something called internal control. Now that's what I said. When patient is positive and we still get negative in the machine, so what is a like how can we say that it is properly negative? Like what is a proof that we have done it properly? So that is where this internal control comes into picture. What we does is, apart from COVID RNA, like target which is present for COVID-19. Along with that, we isolate the RNA. When we are doing RNA extraction, they target for one housekeeping gene. That is usually in COVID-19, they are doing it for RNase P. RNase P is a normal gene they are targeting as an internal control. So this is also isolated along with COVID RNA, which is used as an internal control. So internal control, it has to pass in all the samples, which means we have done the extraction correctly. So even if patient is negative for COVID, he will be having RNase P gene as it is most conserved and housekeeping gene. So IC has to pass. Internal control will amplify, which means we have done the extraction correctly. This is how IC works. If IC fails, internal control fails, then we believe something is wrong with extraction. So we go for repeat extraction. If again IC fails, we could tell there is something wrong with sample. So we ask for repeat sample. This is how it works. Okay. This is all about quality control. So you would ask what are the genes targeted in COVID-19? So usually, as we observe, we have gone through first wave of COVID, second wave of COVID, and we don't know, we might enter again for third wave of COVID. So if you have observed all these waves, we would have listened that, like we would have got to know that there are n number of strains, mutants which are formed in COVID. So if we target a gene which is mutating again and again, it is like early diagnosis becomes very difficult. So usually what they does, whatever primer probes they gave us, so that will be mostly for conserved genes. So it will be same. Even if we get first wave, second wave, the kits will be same, almost same, because the conserved genes are targeted here. So in those conserved genes of COVID, it's nucleocapsid gene, envelope protein gene, RDRP, RNA-dependent RNA polymerase gene, and ORF genes. So these are mostly conserved genes, and in IC, mostly they go for RNase P gene. Yeah. Now we come for last but not least very important topic, that is RT-PCR analysis. Now, if you see that picture, that is a 96 well plate. So previous. This is a 96 well plate which is loaded here. So in RT-PCR, usually this is only mostly preferred. At a shot, we can run 96 samples. This is a amplification plot, how it is seen in the result analysis. So if you observe, it is marked with 3 different steps. Usually it has 3 different phases, I can say. One is background, one is exponential, one is plateau. Now if you observe, like, where is this amplification is starting? From there exponential phase starts. But now I'll start with background. So background is normally, I said initially, like, they have the one in master mix kit, they give one as enzyme mix and one as primer probe mix. So in primer probe, these probes are present, that is Tachman probes, which is having the fluorophore molecule. So we would have added it into all the wells. So obviously, little amount of fluorescence will be seen. That is where background line comes into picture. Little fluorescence is seen almost until 15 cycles, maybe 15 cycles. So till there we would see the background line, and this is not exactly, I will say, exponential phase. Exponential phase, here it is starting, like, x-axis is a cycle number. So it is 40 cycles. With regard to this picture, it is 40 cycles. In y-axis, we'll see the fluorescence. So here, if you observe, where the exponential phase is starting is near the 15th cycle. So in 15th cycle, whatever RNA is present, that is we are targeting for, it starts amplifying. As these primers are bound, so it will start amplifying, and exponential, as everyone knows, it will go on doubling every cycle of the PCR. So that is where exponential phase comes into picture. And at one point of time where all these amount of like products, for example, DNTPs, everything, when it gets over, it stops and it gets saturated. I mean, not saturated, reaction is completed almost. Then it plateaus where the amplification product is only so much, and it shows a straight line after some cycle. And then here, as they said, exponential begins with 15 cycles. That doesn't mean it will start with the same cycle. Usually, we in see RT-PCR analysis, 2 things are very important. One is sigmoidal, and yeah, I forgot to tell you, this graph is called sigmoidal amplification pattern. This is a main thing which is observed in analysis. So there are 2 things where we use for analysis. One is the sigmoidal pattern, sigmoidal curve, and one more is CT value, cycle threshold value. It says the amount of PCR product. So this cycle threshold value is nothing but, if you observe here, I said where exponential phase is starting. So if you observe that amplification is starting around 15th cycle, that is our CT value. So these 2 are observed. So and CT value to say, it is inversely proportional to the viral load. So basically, government provides one cutoff CT value to say whether the patient is positive or negative. And one basic thing is, always CT value is inversely proportional to viral load. If CT value is less, which means viral load is more. With regard to this plot itself, I can say this CT value is 15. It is almost strong positive, which means more the nucleic acid, sooner it starts showing the fluorescence. That's how it is. So it is inversely proportional. The lower the CT value, the more PCR product is present. Next slide. Next slide. Now you can observe. So I said, like, how this positive curves and negative curves will look. So I said initially, IC gene is also seen. So basically, at one RT-PCR analysis, we check for 3 graphs. We get 3 graphs. 3 channels will be there, 3 different fluorescence, 3 channels. So in that 3 channels, each will be for one single target. So usually what I said for 4 genes, those are the genes they will check, but in one single lab, they will go for 2 target genes, and it is completely dependent on master mix kit, what they are using. So whatever the kit insert says, so that is where the target comes into picture, whatever primers they have provided. So here, if you observe in positive curve, there is a one green line which is present. That is a threshold line. Usually, some softwares, what it does is it automatically sets the threshold. In some softwares, we are supposed to set the threshold. And threshold is basically to avoid the background noise. What I said before, background line where there is a least amount of fluorescence, to avoid that, because that is not our product. It's just showing because in all the wells, these Tachman probes are there. So to avoid that kind of background noise, we put this threshold line. Sometimes software does it on its own. And so here you can see, like previous slide. There are 3 genes which are sigmoid. So ORF1AB gene, N gene, and IC. So in 3 channels also we have got sigmoid curves. And if from this only you can make out CT value. Usually in software, it provides us a CT value, but seeing the graph itself, we can make out the CT value. One is around, it's all around 22 to 24. It's all around 24 CT value. So IC is also sigmoid. IC is sigmoid, which means internal control, which is a RNase-P gene, human housekeeping gene. So we have done the extraction perfectly. So, and then if we go to ORF1AB gene and N gene, it is amplified, which means patient is positive for the COVID. This is a positive curve. And next curve, if you observe, that is negative curve, it is saying. That green curve which is come there, it is a IC, internal control. So in negative, to say as it is proper negative, this IC has to pass. So this IC has passed, and other 2 are below the threshold, straight line it comes, which is negative for COVID. Next slide. So now you people can answer which is positive and which is negative. Anyone?
The right one is negative and the left one is positive.
Yeah, that's it. Next slide. Previous slide, ma'am. Sorry. Yeah, it's understood, no? Like all three are sigmoid, and you can check the CT value also. I think it's not so clear. It's taken from the software. It is between 24. It is between 22 to 24 again, all 3. So which is red here, that is one separate, that is IC, which is called Texas red, and other 2 are FAM and HEX, other 2 channels for ORF gene and N gene. Here it is only Texas red for IC. And that red line which is seen is a threshold line to avoid the background noise. This is my team in molecular diagnostics. Actually, everyone are not there. Only few of the staff are there here. She's my doctor, Dr. Sophia.
Which is you in that identify? Because you have not come in the video mode.
That is the one.
Yeah.
Thank you, everyone. Thank you, Hema ma'am, for this opportunity, and thank you one and all present here.
There are few questions which many of them have asked. I'll just tell you. Because COVID has an RNA genome, So in
Yes, ma'am.
RT-PCR, you are going to convert into cDNA?
Yes, ma'am.
Yeah.
Yeah, it's first go for the meshing. In master mix, we have an enzyme mix. In this will be usually you would have used it in college, where you will add DNA polymerase. What is the role of DNA polymerase? It extends. It as a DNTPs. Likewise, to convert RNA into cDNA, it's required that we have reverse transcriptase enzyme, because reverse transcription is happening there. So in enzyme mix, they would have given us a reverse transcriptase with DNA polymerase. So in the machine, first step, which is like they will set a protocol in the machine. For cDNA synthesis, they will set a separate temperature, and other steps also they set a temperature and time. So cDNA synthesis occurs first and then goes for amplification and then for detection.
Yeah, one more question. Why do they use a buffer solution in transfer medium in the swab test?
No. In transfer transfer medium, that is a media, just like media. There is no buffer in that. In that is a media which is having the antibiotic and antifungal agents. Like how we add the antibiotics while preparing the media in college in microbiology cultures. Just like that, here also they add so that other bacterial and fungus doesn't grow in viral culture. Like it's just a we require only virus, so other things that should not grow. That's why we are
Your audio.
adding, like they would have, that is also commercially available. They would have added antibiotics. And hello,
There's a break in your voice, Ashwini.
yeah, are you able to hear, ma'am, now?
Yeah, I'm able to hear you. Yeah, please.
Yes, ma'am. That's what I said. We in VDM, viral transport media, there is no such buffer in that. There is only antibiotics and antifungal agents, and moreover, that is also commercially procured from company. So, whatever antibiotics and antifungal agents they add, it is just to avoid bacterial growth, and antifungal agents to avoid other nucleic acids in that. That's it.
Okay, there is a question where they are asking you to explain about IC again. Was I audible?
I see. Okay. I would tell like this.
Ranjita,
Okay.
focus
So now
the
just
slide,
imagine.
no? Yashaswini.
Hello.
Can we
One
focus that slide? Yeah,
Yes,
that will
ma'am.
be better.
Yes, ma'am. Yes, ma'am. Yes, ma'am. So they're asking how IC works, isn't it, ma'am? Okay, ma'am. Ma'am, you're starting with the slide, or should I start?
One second, Yashaswini. I'll share the screen.
Okay, ma'am.
Is it shared?
Yes, ma'am. It is shared. So yeah, internal control. I can say this positive and negative control understood. It's just about internal control, is it?
Yes.
For example, just imagine we have got the sample and there is no internal control. Just forget about it. We have got the sample and we have done the extraction. As how, like, for example, we'll be doing the extraction in college also. So when we do the extraction, how do we check? We load it onto the gel electrophoresis and check for the bands. But here it is completely graph. So we doesn't know whether we have done the extraction correctly or not. To know that, and moreover, many sample we are getting the samples not only positives, we get negative also. To mainly to eliminate this false negative results, we use internal control. So every human, when he is given his sample, obviously we would have got RNA from human sample also. So what they does is they target a single RNA from a human sample, which is most expressive, like housekeeping genes or most conserved genes, housekeeping gene we can say. So that is a gene. Usually they target for RNase-P gene. Mostly this is a one which is used as internal control. So when we does the extraction, commercially they would have given us a primers for RNase-P gene also. Obviously, if that patient is even negative for COVID, he should be having RNA for RNase-P. So that becomes our internal positive control to check whether we have done extraction correctly or not. So when we does the amplification, even that patient is negative for COVID, he will be having the amplification plot for internal control gene. That's how we can say we have done extraction correctly and he is proper negative. That time only we can give his, we can say he is proper negative. Mostly to eliminate false negative result. I think I answered, ma'am.
A few more questions.
Yes, ma'am.
Pradunya is asking, should point mutation in conserved regions be tolerable since primers have more flexible pairing than ordinary DNA strands?
I didn't get it, ma'am, actually. Can you please repeat it, ma'am?
Yeah, point mutations generally in conserved regions are tolerable since primers have more flexible pairing. Why
To
is it
say
so?
about mutations and with regard to primer and probes, I can't give you more information because that is one master mix thing, and moreover we get the commercial kits where they doesn't mention even to us like what sequence is they are targeting and what is the sequence of
Yeah, there's a discontinuity in your voice, dear.
primer. Even we doesn't know about it. That is a component of master mix.
Okay.
Hello.
So Puama is asking, what is a single seminoid curve which appear in negative result?
Single sigmoid
Curve which appear in negative.
in negative. That is what I told. No, now like to eliminate the false negative result, we require amplification plot in negative also. That is IC, internal control. It is.
Yeah, one Rakshita, she's asking which method is effective, whether it is a swab test or antibody test released by ICMR for testing COVID patients.
Yeah, in this, I would suggest always go for RT-PCR test. And in RT-PCR test, there are some conditions where you have to give sample almost 5 days of incubation of virus, because even if you give before the incubation period, you will get it as negative. And many of the people will, as they get negative and they are still observing the symptoms, they go for CT scan. And CT scan, I would suggest, you will get to know whether there is positive or not, but it is very harmful. Like usually we are not supposed to go for CT scan. And when we come for antigen test and antibody test, both are not sensitive, you can say. So for example, if we go for antigen test, patient is having the symptoms, and sometimes what happens is like internally if he is having the immunity and is fighting his body is fighting towards the disease, then if he is fighting, like antigen, if it is present, it would show positive, which is actually false positive. And if we come to antibody test, It will not come positive initially at all, because antibodies will take time to be produced in the body. It will at least take 6 to 8 days to be produced. So even if you give it before, for antibody test, it is negative, which means it is false negative result. So it
Nowadays
is better.
Nowadays they are doing one more test which they are doing in hospitals, that they can get an instant result within 2 hours, but costing very high, around
Yeah, ma'am.
3,000 to 4,000.
Maybe I think you are telling about Gene Expert.
Is it a Gene Expert or TrueNut?
Not TrueNat, ma'am. It is, I think, CBNat.
Okay.
So it is. This is actually like it is very much enclosed area. There is no need to do extraction in that procedure. Actually, we are also doing the validation of that same instrument right now. So we get the cartridges for different tests. For example, if we are doing COVID, so we'll get the cartridge for COVID and separate software for that, and we just load the sample in that. We just prepare a sample and load the sample and just put the cartridge inside the machine. It will do everything on its own until the result analysis. Even RNA extraction, master mix, everything is, all the components is present inside it, and within 1 hour, 45 minutes to 1 hour, we get the result in that.
Yeah.
And even analysis also, ma'am. In analysis, here in RT-PCR analysis, what we do is we see the sigmoid curve, CT value, and then say whether it is positive or negative. But in that machines, like automatically it will just give COVID-19 detected, COVID-19 not detected. Like that, it will only do the analysis and give such a automated nucleic acid amplification tests.
Okay. So there is a question by Sumana. How can one get into diagnostic field? Will masters in relevant field suffice or we do require other courses?
Is she asking about the diagnostics, molecular diagnostics?
Yes.
Yeah, actually, they consider here when we have the qualification of masters itself. You are will be having better options here. Like as now, if it is observed due to COVID-19, because COVID-19, like if you have observed last in March itself when we got the second wave, it was almost every lab was handling high number of samples. So that one, like we require high number of technicians, even though we have automated extractors. Even to in automated extractors, we have to just load the sample. Even for that, we require technicians. So what they does is they take many number of technicians that time, and if COVID gets over, it's a better opportunity to for all of us who are working in molecular diagnostics, because I would say not only COVID, RT-PCR is not only used for COVID, it is used for many things. Even in our NRL, they use this RT-PCR for many other things. Like one, we can tell for oncology testing. So when there is a mutations seen in cancer patients, those mutations are observed using RT-PCR. For example, BCR-ABL, JAK2 mutations, all these are done using PCR. And when we go to infectious diseases like HIV, HBV, HCV, HPV, and many other tests are there in infectious diseases. And I think every lab will provide us protection in the same way, and it is a very good opportunity to learn here. Actually, you require masters. Usually they ask for masters.
What sector are they very choosy about any specific division?
It depends on the labs, ma'am. Like here, like they're asking about combination or what in masters.
Yeah, masters combination, I'm asking.
No. If even if you have biotech, that's enough actually. MSc in biotech is enough.
Okay. So Pradyumna, she is asking about, is there a possibility of internship and how about job growth?
Job growth is very good. Obviously, as you all know, today it's COVID-19. If not COVID-19, there is something else in future. That is how healthcare industry works. They have one or the other, one after the other. And what else she asked?
One second. About job growth as
Internship.
well. Internships, yes.
Internship. So internship, that depends on the company. So sometimes they may take or sometimes they don't. Usually they go for hiring as a permanent employee. I believe so. I don't know about other companies. Here, whatever I've observed, there is no internship opportunities in such a way. Because this is diagnostics. So internship opportunities are very less.
Sumana, your hand is your query answered, or you have any other question to ask?
No, ma'am.
Azur
That's
is
it, ma'am.
asked.
Thanks to Yashaswini ma'am for answering my question.
Okay, Sumana, thank you.
Thank you.
And Azhar is asking, where have you done the internship and where are you working currently?
Actually, I have not done any internship. I finished my master's in Maharani Lakshmi Ammanni College. As soon as I completed my exams, I got the job opportunity in this same Dr. Lalpath Labs. So I joined as a full-time employee here. So I joined for molecular diagnostics. But apart from molecular diagnostics, they do have other departments here. So I'm working in other departments too.
That's great. Anybody else has any other queries? Are Malbio related jobs in diagnostics?
This is completely molecular biology related. Whatever I told you now, it is completely molecular biology related. Actually, it is molecular biology. That's what I said. Not only you'll be doing RT-PCR for COVID, it is done for many other things. And what I have understood till now, like learning RT-PCR not only in this COVID and other infectious diseases, learning it in oncology panels is very helpful in future. That gives a great career, actually, I believe. If you go into molecular diagnostics department.
Some of them are asking, have you any openings on new Delta plus variants testing?
No, ma'am, no.
Okay. Any other queries? Hope no questions more. I take this opportunity to thank Yashaswini for her wonderful talk and her patience to answer all the questions and all the queries answer. I thank you very much, Yashaswini.
Thank you, ma'am. Thank you very much, ma'am. Thank you for all of you for giving me such a great opportunity. And thanks for all. At the last session, you heard it so patiently, and thanks for all.
Thank you. Thank you so much.
Thank you, ma'am.
Hemavati? Hema? Shall
Yes,
I hand
ma'am.
over the session to Archana or we have to wait for some time?
I think Archana will take over, ma'am. Archana, let you take over. We need to see if the guest has joined.
Okay. Archana?
Yes, ma'am, I'm here. I'll
Yeah.
continue. Thank you, ma'am.
Yeah, please take over. Thank you. Thank you all.
Thank you. Good afternoon, all.
Good afternoon,
Good afternoon, ma'am.
Yeah, so indeed it is really, really, really very happy, and we kind of from the organizing team, we feel very fulfilled, when we see such a crowd. And from day one, okay, from yesterday till today, until the valedictory function, the enthusiasm, the kind of participation which the crowd gave us, the encouragement you gave us was very interesting. See, every teacher today or any time, only thing what we expect is for our students to interact. Only when you understand, only when you're able to go with the topic, you will start interacting. So this crowd, especially from the first talk, okay, not even from the first talk, we actually had kind of ignition, the ignition for learning, the spark for learning from Nagraj sir and Jairaj sir's conversation. If you agree with me, yes, true. So when they started talking about COVID itself, we had questions. I was looking into the message. You all people started interacting. You started to ask questions. You started to get involved into the workshop there itself. Okay, so we had a very good start there. All of us started getting involved into the learning process, and from there, when we had our first speaker coming into picture, she kind of gave a storyline. Yes, right. She taught us PCR in a way that anybody can understand. It is not just a science background person, anybody can understand. Yes, listening to that, they'll start getting in the topic. Same way, the next speaker from Eppendorf, she gave us how the instrumentation works, how we have to go about with it, what kind of mistakes we usually do in it, and what we ignore. Most of the times when I worked, I used to ignore a few things, the variants and all. So those things, where exactly we have to look into, what corrections we should do, everything was excellent. And never the crowd let us down. The organizing team is really thankful to you. The cheerfulness, the kind of interaction from the first talk to this last talk, the question and answers, usually, in some talks we never get questions, we never get queries. We build it up saying, okay, maybe the speaker was very good, she has delivered it in a very good way, so there are no queries. We used to tell like that. But this workshop, we never came into that situation. Thanks to the crowd, thanks to each and every one of you. So with this thankful note, I welcome you all to this valedictory function. So now in this valedictory function, we will have our controller of examination, Dr. Renuka Srihari, who will be addressing the crowd. Welcome the gathering. Later we have our guest of honor from KSCST. So this workshop, we had such huge response from all the audience, such huge response from across the states in India. So that was because of this collaboration and the topic. The topic was a timely topic. We actually planned this very spontaneously, and we also had speakers agreeing to it so spontaneously, and we had very good speakers across. And all the things what was narrated and what was taught to us was delivered very, very, very neatly, and everybody could understand. The crowd has first year BSc students to research scholars. So everybody were interactive. Everybody could understand, go hand in hand. Nobody felt that, okay, this is out of the line. It is tangential. It is going above my head. I'm not understanding. Nobody complained like that. We had excellent feedback from all of you saying the sessions are awesome. You're able to understand. You're going along with the sessions. You're able to grasp the topic. This is how you gave us the response. We were very happy. Thanks to you again. So starting with, we'll have, is Renuka ma'am joined? Ranjita.
Hello, Archana ma'am. Renuka ma'am is yet to join. We'll just wait for 2 minutes. Hemant sir has joined the session.
Okay. So yes, did you all go through that article which Nagraj sir actually mentioned while they were conversing?
Yes.
The COVID article.
Too long.
Yes.
Yes, ma'am.
Please go through. Okay, yesterday I was going through. It is a wonderful article. Wonderful article. It is not that only if you know science, only if you're a person of science, you will understand it. It is just like a comic story. Okay, it just goes like that, and it tells a layman about how this COVID came, how it is ruling the world exactly. So it is a wonderful write-up. Just go through it, okay? If you have not noted the link, I will share the link in our WhatsApp group later. So read that. It is a wonderful write-up, seriously. Even if you have no knowledge about this corona, COVID, PCR, anything, but still you will enjoy, you will be able to analyze things, how things came down to this level, how it turned to be so serious, everything is there. So I request all of you to go through that. Okay.
Archana ma'am, Renuka ma'am is here.
Yeah, okay. So yes, today our validatory session, we have our controller of examination, Dr. Renuka Srihari. She's a person of distinct vision, a fountainhead of illuminating ideas, an idol of knowledge and experience, who's a continuous inspiration to all our teachers as well as our students. So ma'am has been kind of enthusiastic personality, and she has been encouraging all the young faculties and the students to continuously take part in activities, take part in workshops, and keep growing our knowledge and keep growing in whatever field we are in. Okay, so with a small intro, I Renuka Srihari ma'am to address the gathering. Welcome, ma'am.
Yeah, 2 minutes time you give me, ma'am.
Yeah.
Archana, 2 minutes. Okay.
Yes, ma'am. Sure. Fine.
Has the guest entered?
Yes, ma'am. Hemant sir is there.
Yeah, 2 minutes.
Okay, sure, ma'am. So yes, we have 2 minutes. Do I have any participant who is ready to come and share their experience, how it went, how 2 days of workshop? I know it was continuous. Anybody sitting from 10 to 5 or 10 to 3 or something, we will not have continuous interest towards something. We will lose our way in between and all. But I never saw it here in this crowd. So how did you manage? How was the overall experience? Anybody can come up and share your experience from the crowd.
Excuse me, ma'am. Can I say?
Yeah, sure. Yeah, come on.
Ma'am, it was very amazing. It's like I don't have words for it. Honestly speaking, when I joined it, I had a doubt that will I even understand? So many people are coming and such a big personalities are coming. Will I even understand? But I understood everything and it was so good. It gave me a new light towards my career and got me to thinking into it got me a new vision of thinking to have a new career in this field and got me a new knowledge about it. Like the knowledge which I had was not even 1%, but what I got over here was like a complete new vision. And talking about getting bored for sitting 10 to 5 or 10 to 3, that never happened. I don't have to manage anything. The interest only made me to just go through it. So it was amazing, ma'am, like really amazing.
Thank you, Nikita. That was an amazing feedback. Really makes us feel happy, actually. These words are the things which we as organizers carry for a long time. So it encourages us at conducting many more workshops, many more such things, which is going to help you people gain more knowledge, and such timely workshops are going to help us survive the situation. That's the most apt word which I can use. It is kind of being biology, life science students, biology students, somebody in our own relatives coming and asking, what's RT-PCR, how they do the test. See, you should not sit in a sorry figure over there saying, yeah, but it has not come into my syllabus this time. I will learn and let you know. That shouldn't be there. Whatever comes, okay, we should have our own interest to go and explore things. That as a scientist, that habit of going and exploring, going and learning every day will take us to greater heights. So continuously learn. There's no limit, okay? Studying a PhD, taking a postdoc is not a limit for learning, okay? So Abdul Kalam sir and all had 14 PhDs. He still didn't stop learning. He was continuously learning. He was continuously updated until the last date. So that is how he was. So same way, taking such eminent personalities as our example, we should keep growing. So I think Renuka ma'am is ready. Yes, ma'am.
Yeah. Very warm greetings to all. Now, on this occasion of the valedictory session, I can visualize the perspective of the galaxy of organizers, the faculty, the principal, and the management of our college, who are jointly joining their hands along with me in extending a very warm welcome to our guest of honor, Mr. Hemant Kumar, Executive Secretary in Charge, Chief Scientific Officer, KSCST IISc Campus, for consenting to be the guest of honor for this virtual valedictory session for a real-time technique polymerase chain reaction, types and application. I welcome you, sir. I extend my heartfelt welcome to the other collaborating organization, Jain University, Christ University, St. Joseph, KS Hegde Medical Academy, Sri Venkateswara University, Mysore University, Davangere University, SRMN College, Telangana University, Adiyaman College of Engineering, Sri Ramachandra Institute of Higher Education and Research, Central University of Rajasthan. Okay. I'm overwhelmed to welcome the battery of ever-charged faculty to this valedictory program. I'm happy to propose my heartfelt welcome to the galaxy of the students, the faculty, the researchers to this August gathering. Thank you and welcome you all. Yeah, over
Thank
to
you, ma'am.
us. Yeah.
Yes, ma'am. Thank you, ma'am. Okay, so yes, as ma'am said, we have Hemant sir as guest of honor today for our valedictory session. So Hemant sir is currently serving as Executive Secretary in Karnataka State Council of Science and Technology. He joined the council in 1985 as project engineer and has postgraduate degree in civil engineering from Bangalore University. For specializations in which Hemant sir is working currently on is geospatial technologies, spatial data infrastructure, alternate building technology, watershed, and minor irrigation tanks and e-governance. Hemant sir is currently investigating the state Karnataka Natural Data Management System Program, State Spatial Data Infrastructure Project, and WIS program, besides few other projects which he is working on. He has so far investigated more than 25 projects, and also Hemant sir has written many research papers on natural resource management, non-organic solid waste, web geoportal, use of GIS and RS for informed decision making. He has published more than 25 papers across various journals and conference papers. He has also been delivering several keynote and technical talks in the national and international conferences. He's also a member of expert and technical committees, including the Committee of Department of Science and Technology, Government of India, National Biodiversity Authority, Department of Biotechnology, Kerala, Jammu and Kashmir, Odisha, Haryana government, Spatial Data Infrastructure, BBMP, BDA, Minister of Human Resource Development, UAS Bangalore, Advanced Center for Integrated Water Resources Management, A3, Karnataka Watershed Development Department, National Data Registry, BIS, etc. So he has organized and coordinated several international and national workshops and events. So Hemant Kumar sir has also received the Geospatial Excellence Award in the year 2014 by Indian Geospatial Forum for Karnataka State Spatial Data Infrastructure Project. His expertise in geospatial technologies is being utilized by several state or central governments for academic institutions in training and capacity building of the staff, faculty, and student. He aspires to spread the utilization of geospatial technology for decision making and developmental planning. So I welcome sir to address the gathering.
Yeah, good afternoon, and thanks for the introduction. And you're almost bubbling with so much energy. It's difficult to compete with.
Yeah, before sir takes over, Archana, I just want to, I'm very much delighted to extend my welcome to Ms. Prajanya, Project Associate, and Mr. S.N. Jairam, Senior Project Engineer of the Energy Cell. Welcome both of you.
Yeah. Okay. So I think by the look and feel of it, I think the program went very well because as you know, I was speaking to students, and one of the students gave an excellent feedback. I think she was so concise and precise in what she got from the training program. This is what we work for whenever there is some amount of background noise. Can I request all of you to switch off your mic, please? Okay, that's wonderful. So the reaction given by the students is really amazing, because that's what everybody who organizes a webinar or workshop looks for. Am I reaching people? Am I reaching the right content? And the delivery is good? And did the participant understand better? I think all of it seems to me that the students are very content with the 2 days webinar, and it really went well. So now let me come back to the area of work we do here in KSCST. We are neighbors to you, next door to you. When we stay opposite to your institution. Basically, we look at science and technology from our council as a medium to promote the R&D work that gets done in the R&D institutions to the people. Because normally when research institutions or any of us do some work, finally it has to reach someone. I think there has to be some tangible benefit to somebody. Otherwise, all the research that gets done in multiple research institutions goes waste. And also we have a responsibility of social responsibility for scientific community as well. Otherwise, I will not be reaching anyone. So that is precisely our council started with the aim of taking science and technology to the people. That is the real benefits of science and technology to the people. And especially when we started, it was mostly downtrodden and things like that. But the technology doesn't have any differentiation or any classification. Any technology which is good can be used by all kinds of people. So with that in mind, we started, and it's been 45 years since we made the beginning. And as most of you know, that Karnataka state is a very progressive state among all the states in India. Because if you go by the IT, if you go by the BT, or if you go by any technology, Karnataka is leading. In every aspect, it is leading. Unfortunately, we have a very good government, successive governments over a period of time. So science and technology always flourished. From our point of view, being from the funded from both the state and central government, we never got any hindrance from anyone in promoting these technologies. And I'd like to mention a few of them here. One is the use of GIS and remote sensing. That is going on for quite some time, maybe 28 years now. And now the stage has come that these technologies have been used day in and day out by the line department. That is the people working in the government departments. Even the regular annual action plan is being prepared using our technologies. I think government has accepted, endorsed the technologies in full throttle. So we have honestly benefit by the government approach. Otherwise, the kind of work we do without the support of the departments, the government, it's virtually impossible to penetrate. So these technologies have been made a beginning and entered life. So that's why each and every activity that gets done in any department now, it has to be through using GIS and remote sensing, which is quite good. And apart from that, we also have a web geo portal wherein apart from getting the offline data, you can also browse through online and you get what you want. And another thing is that we have a academy industry interaction cell wherein we support engineering students, final year engineering student projects. Normally we support from the past 3 years about 1,000 plus engineering student projects. This project will have 3-4 students. So this way, this year itself we supported about 1,110, 190 projects, which benefits approximately about 5,000 students. Funding to the range of 7,000 to 15,000 depends on the kind of projects they put to us. The biggest advantage is with the council's promotion of this education funding for engineering projects is the merit of this funding. Because these projects get evaluated by the faculty and scientists of Indian Institute of Science and other R&D institutions like NAL, GMT, and so on and so forth. The students get pride in that getting a project sanctioned by KSCST because a good number of people, the quality of evaluation is very good. So we do 3 kinds of evaluation. One is the beginning, to shortlist the project. The second one is for the midterm course correction. If the project is going haywire or the evaluator feels that he needs to offer something during the middle of the project, he does it. And finally, we do the final evaluation to check whether the project has really achieved the proposed deliverables, and they are shortlisted for the best project award. And further, if the faculty is interested, those best projects can be converted into faculty research grant, wherein you fund the faculty also to further fine-tuning the product developed by the students and also make it commercially viable and acceptable. Because we don't want the student projects to end as a student project and writing a report. That's not the end. We want to take it further and see something tangible happens. That can happen only through commercialization. So this is what we expect from all of them. Apart from that, we have the rainwater harvesting. I think rainwater harvesting is here to stay in Bangalore, because now it's been mandatory, and if you don't comply it, you will be charged for it. You need to pay penalty for it. So far, we have claimed penalty close to 6 crores. I think your neighbor is BWSSB. We are working with them. So BWSSB have collected 6 crores this year itself. So that's one of the good sign. And more than 1.5 lakh houses have been have installed rainwater harvester system, thereby storing enormous rainwater, close to 4.4 TMC of water. This is one of the good things. We conduct large number of training programs for engineers, architects, and plumbers. This goes on at least once in 2 months. So we have now engineers and plumbers at the ward level. So if you look at and browse our website, you will get all the information that you need. The plumber's address, name, phone numbers, and all that you get. So that way you can reach them and get your house. If you have not yet done it, you can get it done. So apart from that, we do some sort of a digital heritage. I think digital heritage is comes in the archaeology departments. I think people, you might be aware of it. It is Hampi site, or else Hampi, otherwise Belur, Halibid, or any sites which are there archaeologically. We have 2 sets of things. One is central archaeological sites and state archaeological site, and some world heritage sites also. So all the state archaeological sites have been close to 844 monuments are there. We are digitizing those for restoring and rejuvenation of that, a restoring purpose. And also if something goes wrong with that, you can build a 3D virtual walkthrough so that people can sit at home and watch it at every angle. So you don't have to visit that. I think, but by visiting, you'll get a better feel and you appreciate it better. But however, if you're not able to visit, you can see a 3D model of that virtual walkthrough. You can watch it anywhere on the earth. That's the advantage. So we have done it, and close to 50% work is over now. Probably it will be completed in another year. So you have the entire digital heritage sites of Karnataka will be mapped, and it will have a 3D walkthrough as well. Apart from that, we also map the traditional water harvesting bodies to make sure that nowadays you must understand that all of us, initially we had the surface water bodies, slowly it vanished because the requirement of water keeps growing up each year. And we started digging borewells, and the aquifer also, I think they left with no water. So now we started rejuvenating surface water bodies so that we again start recharging the groundwater and get some additional benefits. So to that level, we mapped all the 14,000 traditional water harvesting bodies, and we have given the government a proposal how to restore it so that it stores water and recharge the groundwater. That's one of the things. And we work on energy cell. Basically, now we are more interested in bioenergy and the solar energy. We provide consultants in a big way, especially for solar energy. So we normally take up consultancy of 1 megawatt power plants. 1 or 2 places is going on right now. Apart from that, bioenergy, I think we are working with Karnataka Biodiversity Board, wherein we promote the use of non-edible oil seeds for blending diesel. And also nowadays, it's more of a sugar industry is coming up in a big way. So ethanol is going to be blended. And further, I think we are also doing a lot of used cooking oil survey in Bangalore. So the FSI, that is the food agency, has come out with an act wherein the commercial oil users have to give back 30% of the oil at the end of the day. That is, if you are buying 100 liters a day, 30% has to be given back, so that will be processed and will be blended with diesel. So blending is taken a big thing, and the government of India has also got a very good, big vision. By 2030, they expect 5% of diesel to be blended and 20% of the petrol to be blended, for which we need to do a lot of work in the sense of technology, in the sense of the quality of this blending, and it should be made available on all the petrol banks. And several petrol banks also now offering that thing, blended petrol and diesel. So this one activity we are supporting, but I think we need to do it in a still bigger way. I hope we achieve the vision and aspirations of the central and state government. Apart from that, housing, I think you must have seen a number of buildings in the institute next to Circle Marama Temple Gate. There are 2-3 buildings which we use. We call it as alternate building technologies. You can also call it as energy efficient building technologies, which we use soil cement blocks, stabilized cement blocks, and things like that. And we have done a different kinds of varieties of roofs using different kinds of materials which are sustainable and economical and cost cutting and also environmentally friendly. We have more than 15 different types of roofing materials and plans which can be used. And in Bangalore alone, we have seen more than 20,000 houses have been constructed using our technology. This is one of the big signs, because for people working on science and technology, if your product or service is used by many people, you're proud of it, saying that, okay, I did something and it benefited someone. So that's the moral of the story. Apart from that, we also have many centers of excellence now. One is on cybersecurity, other one is on aerospace and defense. This is going on, doing very well. And I think now the students require the parent information center. So all the institutions which we're working with, we try to tell them that you have an IPR cell, so that students and faculty know what's IPR and what's the benefit of IPR. We have established more than 40 IPR cells. I think it's intellectual property rights. I urge the college management and especially Dr. Babida, who called me many times to conduct this program. They can talk to the management and have an IPR cell for the students, so that students learn about the science, technology, and innovation as well. So it completes the ecosystem. So IP ecosystem is here to stay, and the government of India and state governments are spending a lot of time and money and efforts to see that IPR becomes part and part of any curriculum, not just engineering and medical college, it's part of the degree colleges as well. So I think there's a lot of benefit, and I hope the colleges also accept and have an IP cell to take it forward. And we do many other programs, like recently we have a cell for deprived sections, wherein we try to take science and technology to scheduled caste and tribal people, the benefits of science and technology that we are doing. And in one project in Yadgir, we are trying to do one project basically which addresses malnutrition, that is stunted growth and things like that, wherein we have the bio dryers, that is dryers for fruits and vegetables. This one project is taking up. And we are also setting up one creation of skill building centers, so that any sort of a skill on sustainable technologies will be given to interested students, faculty, or any interested person, not necessarily the college or institutions. So these are some of the activities which we are working on, and we have been proud to associate with all this. And the last, I would say that it's really a pleasure to work with MLA College, because we are doing this from past quite some time. And every time I get a very positive response from them, and I hope, believe our collaborations continues further. And thank you very much for calling me today and asking me to speak in this webinar. And over to you, Archana. Yeah. Hello, Archana, I can take over from now.
Arjuna has left. Ranjit?
Yes, ma'am. I think there is a technical glitch in between. I think Hema, ma'am, you can take over.
Yeah, I will just take over.
Yes, ma'am.
So
Thank you.
welcome to our guest of honor Mr. Hemant, Executive Secretary, KSCST cell. I also extend a warm gratitude to Dr. Renuka Srihari, CEO, MLAC. So it gives me immense pleasure, to see that students have shown a real interest for our workshop and have participated. And I'm sure that with the feedback that we have got through our WhatsApp chats and interactions and all those things, the response is very overwhelming and very pleasing. I would just like to highlight how this workshop just took its shape. So considering the current COVID pandemic, we realized the buzzword RT-PCR, the test results, positive or negative. Quite scary, but in the scientific community, we felt that, when RT-PCR is gaining so much of importance, are students really aware of what it is, how it works, how the diagnostics, where RT-PCR is involved and has played a major role in the rapid detection of the pathogens. Considering this, and also the aspect that in the curriculum, PCR is only stretched up to the basic principles, process, and the applications, broad applications, we felt that the students need more exposure on PCR. Not many of us are aware on the variants of the standard PCR. What PCR is used for, what type of reactions, on what type of experimental studies it has been employed. Not many of us are really aware of it, including research scholars who are doing PhD and all those things. Without a proper knowledge on PCR, research does not take its shape. So considering all these aspects, the Department of Biotechnology with peer review on this particular thing, we finally took up the 2-day national level workshop. The next target was for us identify the speakers. Whom are we going to select for the speakers? So a quick profile checkup was done to identify the research areas of studies of various scientists and how well they could be useful for this workshop. So that is how all the resource persons for the workshops were identified. So the 2-day national level workshop was then decided to take up its full shape and fully execute under the collaboration of KSCST. We extend our gratitude for accepting our proposal for the benefit of the student and the teaching community. The program was scheduled 28th and 29th of June 2021. Initially, we thought of planning for a 3-day, but later confined to 2-day. The day one program had its beginning with a very small inaugural session by a very known eminent scientist in mycobacterial detection studies, Professor V. Nagaraja from Department of Microbiology and Cell Biology, Indian Institute of Science. In the auspicious presence of Shri K. Jairaj, Managing Trustee, MLA, MLAC, and Professor Shashikala V., Principal of MLAC. The inaugural session had a keynote address by Professor V. Nagaraj, who highlighted on the historical developments that led to the discovery of PCR. He mentioned the contributions of each and every scientist, right from Hargobind Khurana to the Russian scientists, and finally to Carrie Mullis, that led to the discovery of PCR. Shri K. Jairaj addressed the gathering by appreciating students for the interest towards the cutting-edge research happening because of PCR in molecular sciences and genetic engineering, and wished a happy learning for the students. The first lecture session was delivered by Dr. Sudha Lingappa, Director, Aristogene Biosciences Private Limited. It's a well-known company known for marketing PCR reaction products, which is used as a teaching kit in many of the colleges. So she was one of the ideal person to having as a resource person to deal on the introduction to PCR, basic components of PCR, and the steps involved in it. So she briefed detail from the basic aspects to the broad applications of PCR. A very informative session for all the beginners on PCR. Dr. Neha Nair, Regional Manager, Appendov Private Limited, spoke on the instrumentation of thermal cyclers, the basic design that goes into it, the purpose of it, and also highlighted on the various aspects of thermal cyclers available to control various features during PCR process steps. This enhances the efficiency in the yield of the DNA that we all intend towards. A very interactive session and very knowledgeable experience. We all wouldn't have seen thermal cyclers. We wouldn't have heard all the types of the thermal cyclers, the features and all those things. And hence this was found to be very, very fruitful session. The last session of the day was addressed by Dr. Deepthi Saini, founder and CTO of Protein Design Private Limited, who explained each and every aspect of standard PCR, right from its principle, the working use of it, and its applications in various molecular studies. She neatly brought out the differences in each of the PCR that we use and what kind of PCR is used for what kind of experimental studies. The day 2 session was addressed by Dr. Deepthi Saini again on real-time PCR and its applications. Real-time PCR has played a major role in gene expression studies, quantitative studies, polymorphism studies. Deepthi Saini gave each and every explanation of the applications with a beautiful experimental evidences. We are indeed grateful to Madam for having elaborated on all the process and terminologies that are used in real-time PCR. The second session was addressed by renowned scientist, Dr. Utpal S. Tatu from Department of Biochemistry, ISC. He spoke on how the pathogenic diseases, which take up pandemic shapes, are often fall under the category of neglected disease. The pathogens which find two alternative hosts between humans and animals was neatly depicted by Professor Tatu, who emphasized that features of both the host play a very important role in the disease spreading and how the control measures, diagnostic measures are taken both in humans and animals. The last session of the workshop was addressed by Ms. Esishwini from Dr. Lal Path Labs, who gave a detailed view on COVID diagnosis that routinely happens in any diagnostic lab. She emphasized the COVID testing right from the preparation of the sample, maintenance of the sample, processing of the sample for RT-PCR. She also highlighted how the interpretation of the test results are handled, the nuisances that are encountered in announcing the test reports, and how it is overcome. We also had a quiz competition exclusively on PCR, and we are glad that we have 3 winners who have passed through this test. Congratulations to them. We will shortly announce them. I also thank Mr. Hemant sir, who emphasized on the KCST role in encouraging science and scientific temper, both in students and faculty. He showed how students and faculty needs to take up research projects which have a fruitful outcome both in the commercial scale as well as societal relevance. He also stressed how projects are evaluated and what should be the objectives of the project. He also stressed the importance of rainwater harvesting, bioenergy needs, and the solar energy, which can have a safer environment, help create a safer environment. And I would like to inform Sir that we will surely consider your advice for an IPR cell in our institution. And I'm also glad to inform you that in our autonomous syllabus curriculum, we have included IPR as a part of the course curriculum. Surely we will take a step ahead towards the IPR cell, and we would also seek your support and guidance on this. Thank you so much, sir, for accepting our invite and being on this occasion, and also giving us a beautiful platform to organize this workshop. Thank you so much. Over to you, Archana. Archana is joined.
Yes, ma'am. Thank you, Hemavati, ma'am. Yeah. So next we have the most awaited results of our quiz competition. So I request Ms. Ranjita to announce the results of our quiz competition.
Yes, ma'am. Sure. Thank you. Give me a second. I'll just share the screen.
Sure.
Yes. So good evening, one and all. Good evening to the dignitaries, chief guest for the valedictory session, and all my dear participants. Yes, I know this is the time that everyone is waiting for. We hope that this quiz competition remains as a good memory and learning experience to all the contestants. Nevertheless, you win or lose. Yes, to us, everyone is a winner as you battle yourself to step in to participate, to overcome your fear and deliver your performance. Facing challenges with strength, determination, and confidence is what matters. And I think all the participants have done it. So first of all, congratulate on that. So yes, with no further delay, let me take this privilege to announce our prize winners. So yes. So the quiz competition prize winners are: the first prize goes to Shivani Mohan from MS Ramaya College. The second prize is for Meghna Mani from Christ University. And the third prize goes to S. Vishal, Indian Academy Degree College, Autonomous. So for your references, we have also just posted the timings and the marks secured by this prize winners of quiz competition. Thank you, one and all.
Thank you, Ranjita. Congratulations to all the winners. Really, I should mention this. When we were going through the scores and all, many of you, the range itself started from 13 to 23. So it was not that we were feeling actually for how will the first years do. They have competition with research scholars and all. But no, everybody seriously, you had taken it very nicely. So the all the fruits have been borne very, very fruitfully. Thank you so much for that. So next we have the formal vote of thanks by Dr. Babita, Department of Biotechnology, Head of the Department, MLAC. Welcome, ma'am.
Yeah, thank you, Archana. And I think this is the concluding remark, which in every program they expect, that the vote of thanks. But this time I thought that this vote of thanks, let there be an interactive one, because our participants have been so much enthusiastic with all the sessions, with putting various questions and clearing out your queries. So I thought let this just like an interaction, let there be the vote of thanks be. Vote of thanks, as we very well aware that gratitude is the first sign of a thinking, rational creature. Now it's my privilege to propose this vote of thanks. Now this vote of thanks, as I told you, this time it's quite a different one. I thought like the thanks to whom I'm contributing, those are the components of our PCR process. So can anyone just tell me what are the 4 important components required for PCR? Yes, participant, can anyone? You can unmute and talk to me.
Yes, ma'am. Can you hear me?
Yes, Vishal, yes. Please tell me, what are the components of PCR?
So we have the template,
Yes.
the DNA template, and we have the buffer mix,
Okay.
the DNTPs,
Okay.
and the enzyme, Taq polymerase, or any thermostable enzyme.
Okay, so Taq polymerase, okay. And one more important part.
The cofactors, we have magnesium.
Okay, so that is also accessory. Actually, I consider these buffer and the cofactors are all accessories. There is one more, okay, based on which we design various types of PCR.
Primers.
Yes. Yes, yes. I think there are various responses. Yes. So I think what I consider. Yes. So now thermocycler is the PCR title, that is PCR types and application. The workshop title is a thermocycler. Now I am going to put up each one of the component into my vote of thanks so that each one gets its own appreciation. So first and foremost is I would like to include the component of PCR. Okay, so I'm going to include the polymerase enzyme. Now polymerase enzyme in our vote of thanks will be the MLAC management, the MLAC principal, the MLAC IQAC coordinator, the MLAC COE, and last but not the least, the Department of Biotechnology with the vibrant faculties. So each one have their own role of importance here. So I would like to thank our MLAC management for constantly encouraging and supporting our all endeavors. I would also like to thank our MLAC principal, Dr. Shashikala ma'am, who's a motivating person for all of our activities. I would also like to thank our COE, Dr. Renuka ma'am, who's always a supporting pillar. And today, because principal ma'am was not available, she was willingly ready to take up the principal's role. And we have IQAC coordinator, who's again a pivotal role in MLAC institution. So I would like to thank our MLAC IQAC coordinator, Professor Sharmishta Datta ma'am. And last but not the least, our collaborating agency. So our collaborating agency is our esteemed KSCST, Karnataka State Council for Science and Technology. So I really take my appreciation and my supportiveness to KSCST, who have been a supporting pillar for all of our workshops, conferences, for MSc Biofuel project. So for any of the event, I think the KSCST have been a supporting pillar for us. So I would like to thank the Chief Executive Secretary, Mr. Hemant sir, for his constant guidance. And I would also like to thank the KSCST representative, Mr. Jairam, who is the Senior Project Engineer Energy Cell, and Prajanya, who is Project Associate of KSCST. Now, through KSCST, actually, the invitation was spread across in and around India, and we could see the enormous participation which has come through this platform. So I'm very thankful to KSCST, and as Hemant sir expressed that the setting up of IPR cell, yes, we have that in mind, and we are also introducing IPR in our curriculum. We have introduced both in our UG and PG biotechnology syllabus. So we have plan of developing this IPR ecosystem at MLAC. So I would like to thank these components, which is acting as a polymerase enzyme. Now the second part of PCR component is the primer. So they are our speakers, right? So speakers are the who actually shared the information about PCR technique and processes. And we had speakers from industry as well as from various, that is industry and research institution, who enlightened about the PCR process. So they are actually acting as a primer, and with their enormous informative knowledge, you could actually gather much more information about PCR. Then we have the DNTPs, that is the nucleotide, deoxynucleotide. So these are our, that is the Department of Biotechnology, MLAC. So in fact, I would like to thank our organizing secretary, Mrs. Hemavati, who has been pivotal in identifying the title, identifying the speaker. So each and every coordination was made through her. And I would also like to thank equally our workshop coordinators, Archana Preeta and Ranjita, who have also been pivotal in designing the invitation, right from creating the WhatsApp group link. Okay, so whatever the technical part, everything, and even the registration link, everything was coordinated through them. So I thank them, and I also equally thank my department colleagues who were through the moderating all the session and even during inaugural as well as in the valedictory part. So I would also like to thank Dr. Jolita, the PG coordinator, Dr. Sushil Kumar Midda, Dr. Gauri Nilima, Dr. Saraswati, who have been played a major role. And now the last but not the least is our target DNA. So can anyone just tell me who could be identified as target DNA? Yes, good, Yogeshwari. So yes, so the target DNA are our dear participants. So participants, so we had, as Archana was telling, that we had a very great positive response from participants who have been enthralled through the sessions right from 10 o'clock to till 3 o'clock, and who were interactive throughout the session. So it was not just a monotonous learning, but it was an interactive session between the participants and the resource person. So we had participants from our MLAC students who were very enthusiastic to have that learning experience, and we had participants from various colleges in Bangalore. To name a few, we had from Jain University, Christ University, St. Joseph, Maharani's College, MS Ramaya, East West College, Maharani's Cluster University. So it is not that I can leave anybody there. And we had participants from other institutions outside Karnataka and within Karnataka. To name a few, KS Hegde Medical Academy, NITE, Sri Venkateshwara University, Mysore University, Davangere University, SRNMN University, Telangana University, Sri Ramachandra Institute of Higher Education and Research, Central University of Rajasthan. And it is not just the student and the research scholar participants, but we had participants even from faculty fraternity. So we had from Bangalore University, Mangalore University, Davangere, Kristu Jayanti, MVIT, BN College of Engineering. So I would like to thank all the participants for attending this session and making a very good learning experience out of this. So I want the participants to amplify, because the target DNA is going to amplify, and you are going to amplify your learning experience of PCR in your research or in your career part. So I thank one and all for your patient hearing. Thank you.
Thank you, Babita ma'am. Yeah, finally, I take the pleasure of thanking our HOD, Dr. Babita, who was the actual driving force behind this whole workshop. She was the one who actually gave us the idea. Yes, this is a good topic. We have to go about. Come on, do it. All of us were actually all getting vaccinated. The pandemic situation around the society, everything, we were actually low. But the actual force which actually gave us the whole enthu to conduct and such a huge crowd responding to it was through Babita ma'am. And thank you so much, ma'am. Thanks for the opportunity. Thanks for this workshop. Thank you. Thanks to each one of you. Thanks to every participant. It was really, really a pleasure meeting you all and interacting with all of you. So hope you all are a part of our future endeavors also. Thank you again. Thank you, guys.
Archana ma'am, photo session.
Yes, ma'am, photo session. So I request all of you to just go on video mode so that we click a photo, have one memory for lifetime. Okay?
Dear participants and the dignitaries, I request everyone to on your video.
Participants, students, come on, unmute your videos. Let us have a pic. Thank you. Yep. Thanks to everyone, guys. Thank you all.
Archana, shall we staff remain for a staff group photo, department photo? I think Hemant sir also can